Okay, so welcome to Denver, everybody. What a fantastic group. I was looking at the attendee list this morning, and between our virtual and our in-person groups, I couldn't imagine reaching a better swath of the community. And so I really appreciate everyone being here, especially the Friday before Thanksgiving. I'm Andy Kiliansky, a program manager here at ARPA-H and the program manager running the APECx program. What we're going to do is we're going to have a bunch of presentations here in the morning sessions about the process, what APECx is, how you submit work proposals to us, and then what we're looking for. And then we also have some presentations from some US government and enablers who are bringing capabilities who are going to be accessible to the proposers for APECx. And then in the afternoon, we have poster sessions and sidebar meetings. For those of you who are able to do an in-person sidebar, we have a schedule out that Nancy has at the front desk. Unfortunately, we couldn't meet with everyone today, and so we do have virtual options, and we'll be communicating with everyone about how we can get some time next week together. Without further ado, I'd like to introduce my boss, Dr. Amy Jenkins, who's the director for the Health Science Future Mission Office.
Good morning, everybody. Thank you all for being here. It's great to see so many faces, some familiar faces, new faces. I'm just going to talk very briefly this morning. You all are here to hear about APECx, but I do want to welcome you to ARPA-H and introduce you to ARPA-H as a whole and Health Science Futures Office. This is an opportunity for us to really make sure that the community understands what we're doing at ARPA-H and how we're different than other organizations that you may be familiar with. All right, so at ARPA-H, we have a very simple mission statement that is to accelerate better health outcomes for all. It's a very punchy mission statement. We take it very seriously. And we want to ensure that everything that we do, we might be inventing technologies of the next 20 or 30 years, but we want when those are in the hands of the customer and in the hands of patients and caregivers, that they are actually improving health outcomes. So what is ARPA-H? ARPA-H was stood up by President Biden back in March of 2022. We have our first inaugural director, Dr. Renee Weigersen. She started in the fall in October of 2022. So we've had her for just over a year now. And we've just really gotten up and running in this past year. President Biden stood up the organization in the business image of DARPA. For those of you who are familiar with DARPA, and many of us here at ARPA-H came from DARPA to bring that mindset. The idea of what DARPA did in the last 60 years for national security and really pushing the boundaries of what we can do from a high-risk technology perspective in national security. Let's take that model and put it towards healthcare. And so that's what we're aiming to do here at ARPA-H. For those of you who are not as familiar with an ARPA or with DARPA, there is a lot of promise in this model. We fund a lot of things that may not work, but we're willing to take those crazy risks. And many times we're shooting so high that we might miss, but every once in a while when you hit, you kind of change the world. So here's some examples of some of the technologies that came out of DARPA. So DARPA was started in response to the Sputnik launch. And many of the things that I think many people here may know, the ARPANET, so early incarnations of the internet, stealth technology, self-driving cars, and some of the technologies around the mRNA COVID vaccines came out of initiatives at DARPA. So willing to put some money behind something. And I will tell you that nobody at DARPA thought they were inventing the internet when they started that effort. Even further than that, it was much different than that. They actually just needed a widget to do something, and it was the foundations for the internet. So oftentimes some of the foundations that we're laying here at an ARPA blossom into the future. So a lot of people do ask this question, so I'd like to put this up. Where is ARPA-H in the organization of the US government? And so ARPA-H is administratively linked to the NIH. We are not an institute or a center or an agency within the NIH. Our director, Renee Weigersen, reports directly to the Health and Human Services Secretary, Secretary Becerra. We do not have any research labs, so we don't do research. You all do the research. We pay you to do the research. We do have funding. As a funding agency, it's really important that you have appropriations. So we have $2.5 billion in appropriations. And for those of you who have read the APECx BAA, which you're going to hear us say this a lot today, read the BAA, read the BAA. For those of you who have read it, you will see that we are not issuing grants. We typically do not issue grants. We issue contracts and cooperative agreements, OTAs. This allows the government to have a little bit more day-to-day say in the progress and the capabilities that we're trying to build. So we have 4 initial mission offices here at ARPA-H. The other 3 are grayed out, not because they're not important, but because we're all here today for the Health Science Futures office. So I'll just quickly walk you through these. Health Science Futures, the office that Andy is in. This is the office where we are, for those of you who are familiar with DARPA, it's kind of like the DSO of DARPA. It's the ARPA's ARPA. And so what we're trying to do here is really expand what's technically possible, thinking about those technologies, tools, and platforms that will really enable healthcare of the future. The Scalable Solutions Office, this is an office in which we're developing technologies to scale. How can we reach every last person in this country, whether they're in rural parts of the country or they're in large cities? Proactive health, pretty straightforward. How can we prevent people from becoming patients? And then finally, the Resilient Systems Office, where we are building integrated healthcare systems. Now you might say like, well, why isn't APECx in the proactive health office? Well, there's gray lines here, and we are not as stovepiped at ARPA-H as we would have been at DARPA. And so there is certainly overlap, but this is a platform program, and we're really excited about the future of this program in our office. And so who do we work with at ARPA-H? We have a fairly large stakeholder community. So we work very closely with our NIH colleagues, and several of them are here today, and you're going to hear from some of them throughout the day today. We work with federal partners like the FDA and CMS, but we also work with private sector, private investors, NGOs, and then maybe most importantly, probably most importantly for today, certainly most importantly for today, are the performers. We fund academia, industry. We get a lot of these questions. What about new professors? What about small companies? What about big established labs? What about pharmaceutical companies? We fund all of the above and can put agreements in place with all of the above. We also take our customers very seriously, and we have a very large customer base. We're here to serve all Americans. And so we take that very seriously. We have opportunities for engagements, and we will have future opportunities for patients to understand what we're doing at ARPA-H as well. So a little bit more about the Health Science Futures Office. As I mentioned, here's where we're going to develop the tools, technologies, and platforms to expand what's technically possible. So these could be anything from the molecular, like new delivery systems, new ways to modulate the host, all the way up to the tissue or cellular level, and then even some things that are maybe relevant at the individual scale. And I'll talk in a few minutes just about a couple of our first initial programs, so you can get a little bit of a flavor of the types of things we're trying to do in this office. So 2 of our first programs here, the first one where we really invested heavily in what we can do around tissue and cellular treatments is the Nitro program. So what if we could make joints heal themselves? This is run by one of Andy's colleagues, Dr. Ross Urich. And then the PSI program, Precision Surgical Interventions, what if surgeries could fix problems flawlessly the first time? And this is run by Dr. Alana Hanku, another one of Andy's colleagues, and really addressing more of that. What are we going to do at the patient, at the human, at the body scale, if you will? So you have these 2 programs that are addressing tissue, body scale, and now we have Andy here directing the APECx program, where we're really investing in molecular technologies and platform approaches. I do want to talk just very briefly, because you'll hear rumblings about this kind of thing, about the model, about the ARPA model and what makes this different. So a little bit of really what makes us fundamentally different is that the program managers and all of the technical government people here are term limited. So we are limited to 3 years terms with the opportunity to re-up for another 3 years for a maximum of 6 years. Andy has to leave in 6 years. I have to leave in 6 years. All program managers have to leave in 6 years, maximally. The reason for that is that it allows us to keep new ideas continually coming into the agency. So we're an ideas agency, we're a projects agency. We run a project, we're in and we're out. At the end of the funding of that project, there is no opportunity to re-up the funding. We're done. We are going to, as you're going to hear later today, ensure that these technologies can make it into the hands of end users through commercialization efforts. And we've stood up a whole office that you're going to hear about to ensure that that happens, but we don't have opportunities for re-upping of grants. But these programs that the program managers come in, they typically run anywhere, they'll start anywhere between 2 and 4 programs, and those will run anywhere from 2 to 4 years, often 2 to 5 years here. And so program managers will manage programs. And then if you're doing your math, they'll leave and they'll take those programs and they'll hand them off to a new program manager who will look at those programs with a new eye, may change some things a bit, but it really keeps the agency constantly thinking about new ideas. And these program managers can put exquisite attention into these efforts. And we want these efforts to change. We want the agency to change. And we believe that this model allows us to occasionally make those crazy solutions like what you saw on one of those first slides around stealth and self-driving cars. And so a little bit on how we got here and how Andy got here. So program managers come to ARPA-H, they come here with a big idea. They pitch their idea to me and to agency leadership. And we say, yes, we like that idea. You should come here. You should be a program manager. Once they're here, they spend some time talking to the community, particularly our government partners, and they develop that very specific program around a specific challenge in healthcare. Today, you all know what we're here for. They launch the program. So today is the launch of the APECx program. And then when you all submit proposals, Andy will evaluate them along with some government reviewers and choose the teams that are going to try to address this program. There often are multiple teams. We do expect multiple teams to be working on a program. And then that program will be managed in conjunction with all of the folks that are working on it with Andy for the next 5 years. And so these performers are, sometimes it won't work. We understand that it won't work. There's lots of opportunity for modifying statements of work, for saying, okay, we tried this approach isn't working. Let's try a different approach. That's really important at an ARPA. We don't spend years beating our head against a wall trying one experiment that just won't work. We just cut bait and move on to the next potential approach. And every once in a while, we're successful. And that's what we're hoping, that somebody in this room or online is going to be, or maybe multiple groups in this room or online are going to be the teams that are successful and meet the end goals of the APECx program. So with that, I wanted to give that brief introduction. I'm going to stop there. I'm going to turn it over to Andy so that he can tell you exactly what he wants. This is an opportunity for you to hear it from him. But I'm going to leave you with final parting words. You all are going to want to talk to Andy and his team today, and that's really important. But the most important thing you can do today is talk to each other, okay? Because I don't think that any one team in this room can solve this problem. It's a very large problem, and you all, having read the BAA, I'm sure, recognize that like there's disparate communities that are going to have to come together to solve this problem. So please talk to each other, go to the poster sessions, see what you need, what gaps you have to fill, and establish those relationships. This is what makes an ARPA special this day. Everybody always asks me like, wow, you all must be brilliant geniuses inside these buildings. No, this meeting right here is how we often get these projects across the finish line, because we put the right people together to solve these problems. So go out, talk to each other, and bring us forward a great solution. So thank you, everybody. I'm going to hand it over to Andy.
Okay, great. Thank you, Amy. And Amy really framed, I think, the purpose of this day really well, right? It's for all of you to talk and to meet and to see what kind of cutting edge innovations are happening at the poster session, right? And think about, okay, if we get a team of, academic PIs and, small startup industry folks, and there might be some computing technology we want to bring in. And that's really kind of the secret sauce for what we're looking for in terms of APECx. And so I'm going to spend a few minutes just walking through a lot of the content that's in the BAA, and in many ways, the BAA is more complete than what I'm going to describe here. So please read that, and we are available today and in the future to answer any of the technical details within that proposal. Okay, so I don't really have to belabor this point to this audience, but the goal of APECx is really to eliminate viruses as health threats in the future, right? And we're going to start to get down that path by building vaccines that are broadly effective across multiple viruses within a genus, multiple genuses, or subfamilies. Right, so we know that there's over 270 viruses that infect humans. We know about half of them cause some sort of disease that we're able to detect. But as we're all aware, we have vaccines and tools to combat these viruses for less than 10%. And more we're seeing, right, correlation of typically thought of as benign or acute infections with chronic illness and later in life disease. And so, the more we're discovering about how we interact with viruses, I think the more we're going to find that viral infection at any level is probably not a good thing. So in addition to sort of the unknowns that are out there, future pandemic, potential pathogens, the next SARS-CoV-2, et cetera, we have a lot of regular disease burden here in the US that are caused by viruses that we don't have a good answer for. In this slide, we've got autoimmune and neurologic diseases caused by herpes viruses, right? And the large burdens we have here in the US related to those infections. We also know that viruses cause a variety of cancers, right? And in some cases, viruses like Epstein-Barr, which we don't have a vaccine for, cause both neurologic disease, chronic illness potentially associated with things like MS, dementia, and Alzheimer's, right? But also cause cancers. So we are very interested in combating the natural evolution of these pathogens, right? With the antigens and vaccines and tools that are going to come out of the APECx program. So, right, there's a variety of, technical challenges in the current state of the art that haven't allowed for us to realize this goal of true universal vaccine development. We were very fortunate that, the scientific community was looking at SARS-CoV-1 for about 15 years before SARS-CoV-2 emerged. And that, body of structural data and experimental data on the SARS-CoV-1 spike allowed for us to understand how to develop a great antigen for SARS-CoV-2 and turn that around very quickly. Regardless, right, it still took 11 months to get those vaccines licensed through an EUA. And at that point, over 1.8 million people died around the world, right? So even though Operation Warp Speed and the US government successes in that space were really impactful, right, not impactful enough. We're seeing the booster uptake in the current way we're vaccinating against SARS-CoV-2. We're losing the attention span of the population here in the US, right? So this data is a few months old, but the updated bivalent booster dose uptake was only 17% around the country. And while we had really great success in that primary series during the early stages of the pandemic, the uptake of boosters, right, and repeated vaccinations, we've seen that wane over time. One of the things that I'm really passionate about is alternate delivery routes, right? Ways to make vaccination more acceptable for people here in the US and internationally. And we're going to talk more about this in later sessions, but we need to be looking beyond the needle, right? So how can we pair really innovative antigen discovery efforts with alternate delivery technologies from the beginning? As someone who previously developed vaccines out in the NGO world, you don't go backwards, right? And so really incorporating these things up front in the development pipeline are really important. And then finally, some of the successes we have had in making multivalent vaccines are 100% successful. So the current HPV vaccines, for example, that we often point to as an exemplar for multivalent vaccine development, they don't cover all of the strains that are circulating, right? So we still have about 10% of the HPV strains that aren't covered by the vaccine that we know cause cancer. And as a result, we have to spend $6.6 billion still in annual cervical cancer screenings as a result. So how can we, within APECx, right, solve these different state-of-the-art technical challenges that we have today? Right, and our goal, going back to that initial picture, right, is taking off large swaths of the viral ecosystem that we understand today off the table from a threat perspective. So how can we look forward into the future, right, by building things at a genus, subfamily, and family level that will insulate us from future evolution, right, and future emergence of infectious diseases, as well as natural evolution happening with viruses that are circulating within our bodies every day. Okay, so again, this crowd doesn't need any, updating on why can't we do this already, but in terms of the computational tools that are emerging, we really lack a few of the foundational pieces we need to really purpose develop antigens for a specific class of viruses. We don't have a lot of foundational protein structure data, which challenges us in terms of machine learning and the algorithms that are being developed specifically for viral vaccine development. We also lack a lot of the prediction and modeling tools to look at things like protein-protein interaction. What do point mutations do within a viral antigen that might escape a vaccine or a therapeutic? Post-translational modifications we know are really important for immunogenicity of proteins, right? But we don't have great ways to model that today. And so discoveries that we're going to make in APECx, right, from a data perspective as well as from a tool perspective, are going to be impactful for what our objective here is, which is broadly effective virus vaccine antigen design. But these tools are applicable in everything in the life sciences. If we can really capture how to best answer some of these questions through the discoveries we're going to make in APECx, we'll have impacts way beyond vaccine development. Okay, so the APECx approach, we have a two-phased approach to the program. Phase 1 is 36 months, and that incorporates 3 core technical areas. So technical area 1 is high throughput analysis, and we'll talk more about these. We'll break them out in a few slides. Tech area 2 is model development and the computational pieces of the discoveries we're making here. And then tech area 3 is translation, right? So that's bringing those considerations like delivery, manufacturability, 1 dose instead of 2, et cetera, right into that discussion from the beginning. We also have a separate track, and so we're asking proposer teams to encompass either all 3 technical areas in a proposal or only TA2. And those TA2 only submitters are going to be focused on evaluating the tools that are being developed across the performer teams. Right. So one of the things that we're really mindful of, especially in computational design and model development, is you can't purpose build a model that only works for your thing. Right. So we want to ensure that everything that's being built and discovered in the TA1, 2, and 3 is able to be stitched together, is able to be kind of at the end of this program, maybe Frankenstein and put together into a design and development pipeline that work for all viral families. And so we're very cognizant of the fact that we need to be stitching these things together and evaluating these models and data in real time. So that's the first 36 months. And at the end of that 36 months, there's a set of metrics that we're asking the developers to meet. And we'll talk about those, but they're also outlined in detail within the BAA. After that phase 1, there is a down selection in which we will take a few of the most promising candidates into phase 2. And that's a 24-month phase. And that is kind of confusingly, that's our phase 1 clinical phase of the program. And so really for us, like Amy mentioned, in Health Science Futures, we're about the tools, we're about the platforms. And so for me as a program manager, I'm very interested, right, in the potential candidates that come out of our phase 1 into phase 2. But really for me, those are technology demonstrations, right? Those are proving out the concept that the tools that we've developed in phase 1 are going to work for vaccine design. And so in phase 2, we will do some human clinical trial evaluation in a phase 1 setting. And we're going to hear from our partnerships and transition office, Patio, here in a few minutes. We've brought on a bunch of services within ARPA-H that we can provide to performers, right, to help with things like regulatory, like manufacturing, like commercial strategy, right? Because at the end of the day, we want these technologies, these tools, these candidates to live on their own outside of government investment. So I kind of covered this already, right? But there's specific technologies and things that we're interested in, specific approaches within these different technical areas. I'll talk to the metrics here in a second. And like I mentioned, the discovery pipeline development, which we're going to be running through a TA2 only set of performers, right? That's data repository and sharing, design pipelines, data curation, evaluating model successes in different settings, et cetera. So we're envisioning teams that are going after all 3 technical areas together, as well as a TA2 only team that sits above and helps integrate all of those data and models across the program. Another unique thing we're doing here in APECx is we're not just funding efforts out in industry and academia. And so we've brought on a great set of US government and US government adjacent enablers for performers within APECx to leverage throughout the lifespan of their project and the program. So you're going to hear from NIAID, both extramural and intramural research today. You're also going to hear from some of the Department of Energy laboratories who have offered to bring things like compute time and model development resources and time on the beamline and things to the APECx performers in a way that will allow for us to go really quickly. Okay, so this kind of reiterates some of what we discussed already, right? And this is kind of the life cycle of the program. And so we're very interested, right, in surviving in the wild. And so we want our models and tools to be applicable broadly, right? So TA2 only performers are going to help there. And then we also want these candidates to be effective technology demonstrations of those tools out in a clinical setting. Right, so we have these 2 tracks for proposers to submit, TA2 only, and there's a separate set of metrics involved in those proposals, and then technical areas 1 through 3. So this gives a little bit of additional detail on sort of the execution in time and space. In the BAA, we have things broken out by year, and so there are some specific milestones and deliverables that we're looking for across the life cycles of these projects within APECx. And then, like I mentioned, at that 36-month time point, we're going to be asking the teams to be essentially evaluating against the rest of the performers within APECx, how effective a vaccine might be in vivo against multiple viruses at once. And then we will take a few teams forward right into phase 2, where we evaluate from a clinical perspective. Okay, so now I'm going to dive a little bit deeper into some of the metrics behind the technical areas. So for TA1, right, the goal of technical area 1 is really high throughput functional data, viral protein structural data, right, and novel methods that will allow for us to generate this data rapidly and in a way that's standardized across the field. So in terms of high throughput analytics in TA1, we've broken out in phase 1 really what we're looking for in years 1, 2, and 3, right? So that's everything from delivering to the government a workflow to really rapidly from a functional and structural perspective, evaluate potential antigen candidates, to everything like how are we going to produce these at scale and really make them available for the proof of concept studies towards the end of phase one. And one of the kind of side benefits of this approach for APECx is we really want to fill out some of the data gaps that we have in terms of understanding virus protein structure, viral antigen design, et cetera. So those are kind of the TA1 metrics. And like I mentioned, the BAA, which the final version published just yesterday, has this in excruciating detail in some cases. And so I encourage everyone to take a look at that. TA2. So these are for the teams that are including TA1, 2, and 3. Right here is the tool design space. Right, so we are striving for a certain level of structure prediction accuracy in the tools that are being developed. In addition to including things like post-translational modification, protein-protein interaction, protein complex formation. We know some of the antigens we might select in our vaccine programs, right, are multimeric, and might be dimers and trimers and multimers right on the surface of a viral protein. And so figuring out ways to structure, model these things in sort of 3-dimensional and 4-dimensional space, really important. And then in phase 2 of TA2, in years 4 and 5, we are anticipating some additional funding to, again, get these tools out into the wild, publish all of that data, publish things into the databases in a way that's queryable and usable for the entire field. So in terms of the teams that submit to only TA2, right, there's some specific metrics, and really that's an integration function and an evaluation function. And so we want the TA2 only submitters to be able to work across the teams of performers across all of APECx, right, to figure out data standards, data storage and curation that teams can have access to, model scoring and evaluation throughout the program as we bring in different pieces and add-ons to those models as they're being developed. And then finally, a way to help us, again, have these tools live out in the wild beyond APECx so that they're accessible to the entire development community. So TA3, right, is the more kind of traditional vaccine development technical area, although we are very interested in alternative delivery methodologies, alternative manufacturing approaches, et cetera. And so in this, we are really, focused on TA3 being there from the beginning. And so these sorts of translational questions and challenges that come up during the course of a vaccine development program are addressed as early as possible. Right, and TA3 is going to be heavily involved in the evaluation of these candidates, both in vivo models and in humans in phase 2 of the program. Okay, so we're going to hear from Amanda later today on some of the specific equity considerations that we're building into the APECx program. But from someone who developed vaccines in the NGO world, right, we often thought of equity and accessibility from the international perspective. We want to bring in those thoughts into this program, but we also, as we're building tools, right, that we want Americans to utilize at the end of the day, we want to be thinking about ways to make these candidates, these tools more accessible to Americans. And not just accessible, but acceptable to Americans as well. And so we are, in parallel to some of the development work, we're going to be doing some research and development in what does an acceptable vaccine look like for the US? How can we really overcome some of these challenges that we have in vaccine hesitancy and using things like artificial intelligence, right, to design future candidates. So we're going to be doing some of that work, and it's possible there will be injects throughout the program based on the findings that we generate from that acceptability research that we're going to be doing. But certainly data accessibility, tool accessibility are other ways we're addressing the equity considerations as well. We want all of the data and the tools that we're developing to be as open source as possible. And we have a couple other initiatives underway at ARPA-H to really tie biomedical data together in a way that's accessible, queryable, and modelable from the tools that we have in-house and with performers at ARPA-H. So there'll be certainly some crossovers between the data and the tools that we're developing and the infrastructure that we're building more broadly in the US government. Okay, so if we're successful, right, I'd like to imagine a future without viral disease, right? And so we're going to start to get there through the APECx program by really pushing the boundaries of broadly effective and immunogenic vaccines in ways, delivery technologies, manufacturing technologies, right, that don't exist today to get to where we need to go. And really that toolkit, the model development pipeline, and making that accessible and really tunable for a certain vaccine approach that one might take. If we can get to that, and that's been demonstrated through, effective phase 1 studies in humans, it'll be a resounding success in APECx if we can build those tools that are usable by everybody to get to a place where we can design vaccines that work against everything. So with that, I am going to turn it over to Jennifer Mack, who's from our Business Innovation Division, who's going to talk in a little bit more detail about the acquisition details of the program. So, what is our R&D solicitation? How do performers work with us? What are we looking for? How do you check all the boxes, cross T's and dot I's to get a successful proposal in front of us?
Perfect. Thank you, Andy. So I am Jennifer Mack. I am a contracting and agreement officer that is supporting ARPA-H. So I am one of those people. I came from DARPA, so I apologize if I go off on some DoD type terms. So please just raise your hand, tell me, maybe pivot back to the ARPA-H world, right? So before I got started, I kind of wanted to know who was in the audience, who my audience is, so that I'm not rambling on about a bunch of stuff that maybe you already know or you're less interested in hearing about today, because this is really about you and how to help you be more successful in proposing. So has anybody not put together a proposal in support of a government agency before, like an ARPA at all? So you guys are all pretty familiar with the process a little bit. So maybe a couple new. So right, NIH, but are you more in the grant world that you're used to? Are you guys used to cooperative agreements? Are you used to other transactions, procurement contracts? Okay, so see some nods here and there. So, all right, that helps me. And I apologize online. I'm not seeing your reactions as well, but I will try to talk to the whole entire group here. So just a little bit, I think that most of you then are aware of what BAAs are. So I will just say that they're a merit-based instrument. And that really means that we, at the end of the day, we're looking for the best solution, right? Do we care about price? Certainly we care about it, but we're actually more interested in saving the world, helping Americans, right? That is what we're really interested in doing. And so, yes, the price of your solutions should be commensurate with what you are actually proposing, right? But your actual solution is what we are greatly interested in. So some of the other little aspects of the BAA that are great for us to use is because that we're not just looking for one singular solution, right? We are looking for a group of performers here in order to come out and, put together an amazing program and come up with some amazing solutions to these problems. So, and I kind of already touched on it, but one of the aspects of the BAA is that we can award multiple different instruments. So historically, procurement contracts, grants, cooperative agreements, other transactions, all of these have been used underneath the BAA. So any questions so far? Perfect. Okay. So some of these award types that will be used underneath the APECx program are cooperative agreements and other transactions. That means that right down here, right underneath the traditional awards, we will not be doing procurement contracts or grants. And Amy already touched on that a little bit, right? For one, we're trying to stay away from the FAR-based world, and I'll talk about that a little bit different later on when we go over other transactions. But as grants are sometimes, or no, they are instruments that we provide you money, you go do your research, and then you publish your results, right? We intend to be more involved in your research, so hence we won't be doing a grant, right? So award funding here, to make sure I did all that as well. So we have not published a budget for this program. And so the most important thing I can tell you, right, is something that I mentioned before. Your solution should be aligned with, or rather your price should be aligned with your solution, right? So when you're filling out cost or price solution templates, right, you should always be thinking about what have I actually said within my technical solution and how do I actually cost that out, okay? So don't be looking for some like grand definitive, budget line that you should be meeting, right? Like actually propose what you have written to, Okay, hopefully that's helpful. And I think, we put these up here, but I do want to say sometimes these timelines flex, right? So I do want to make sure that at all times you are looking at sam.gov to make sure that these timelines that we posted up here, especially when you should actually be submitting your proposal, you should be looking at the BAA, you should be constantly refreshing sam.gov to make sure that these timelines don't switch. So I will say that the government hopes to get into negotiations in April 2024. That does not mean that we are absolutely committed to that date, okay? So please, again, you'll be constantly updated. ARPA-H is doing an amazing job with that, making sure that you're up to date on all information. So I will not belabor that point anymore. Okay, so let's go into cooperative agreements. So those of you who are not aware of cooperative agreements, I will say traditionally, I have only awarded these instruments to universities. That does not say that they are the only ones that can receive them, but I will say that that's what I'm used to, okay? Why I generally award them to universities is because a lot of their research is then published to the public, right? And that's an important distinction there, because even if you are a university, if you are going to do research and then place restrictions upon your research, right, it is no longer able to be published to the government in totality, right? So I want you to think about that. If you are only doing what I used to say at DARPA is that basic research, right, that theoretical research, right, you will be more in the realm of this cooperative agreement instrument, okay? Again, traditionally, they're provided to universities. So I just wanted to hit on that point because on some of the other programs that I'm doing, many questions come in, should I do other transactions? Should I do cooperative agreement? And I will say, if you have IP restrictions in any particular way, the cooperative agreement is not going to be the best instrument, okay? It is governed by a whole lot of different laws and regulations. And again, talked to it a little bit before, is that substantial involvement is what we mean by being involved in the program. And that's why we do not provide a grant, right? We go with the cooperative agreement. Okay, other transactions. So anybody just don't know what these instruments are whatsoever, another transaction. Okay, good. All right, so another transaction is, how do I put it in common frame, is like, when you sign an agreement, right, with a phone carrier, right? They have this entire agreement that you have to agree to. You probably don't read any of it, but there are actually a bunch of articles in there telling you, and generally you can't negotiate with them. But anyway, you sign up and you agree to it, right? So that is not what this contract is, right? This agreement is. Rather, another transaction is filled with a bunch of articles that talk to how we can create a win-win solution between you and the government. And that means that all of those articles within that other transaction are negotiable, right? Now, every agreement officer is different. Every solution is different. Every situation is different. So what we negotiate, what the government is willing to negotiate may differ from project to project, program to program, agreement officer to agreement officer. So be mindful of that. Just because you received and were able to negotiate an article underneath one other transaction doesn't mean that you'll be able to do the same thing underneath the next other transaction, okay? But the basis behind it is that we are trying to do business with you guys much differently than what we historically have done in procurement contracts, right? And procurement contracts are governed by an enormous amount of regulations that you have to follow, that I have to follow. And not to say other transactions aren't governed in some way, they are by statutes, but they give us the opportunity to meet you in a better place. Okay, so what I guess I want you to take away is that They are made to form a better relationship with you. We are trying to create a win-win solution. We know that you're not trying to manipulate us, and we're not trying to manipulate you. And so this is our way of creating a better relationship with you guys. Okay, so throughout the day, if you have more conversations, and please read the BAA because there'll be additional information in there. We can discuss more throughout the day. Okay. All right, so here are just some pros and cons for each instrument. I mentioned some of these, so the other transaction right is not governed by an enormous amount of regulations that you'll find in the FAR. And then, right, intellectual property, I spoke about that in regards to cooperative agreements, right? But this is where intellectual property does come in, right? We understand that your IP is important to you. And so we want to have those real valid negotiation points and understand, hey, what does the government need and what do you need? And how can we both win throughout that negotiation process? I don't have a lot of cons with other transactions other than sometimes if you want to negotiate a lot, they take time, right? And so that is the one thing, they are not necessarily a fast solution. They can be if you accept what is written and provided to you. But the more that you want to negotiate, the more you get lawyers involved, the more people you get involved, right? Yeah, they can take some time to negotiate. Okay. Cooperative agreements, spoke to these a little bit as well, but in terms of like the pros, you know, for some of you who are universities, right, this is very well-established system that you have in place. You have rate agreements, you have everything, and it's, you know, essentially the PI writing out the technical solution, and then some of this is already done for you, right? But some of the cons are, right, is that, right, those lines are very much drawn for you, okay? So, but I will say, the cooperative agreement is the most important or most appropriate rather, if you are doing that really theoretical research. Okay, great. Let's get into some of the specifics with the program. So in this program, APECx, right, you do have to submit an abstract submission. That is mandatory, and your abstract has to be selected in order for you to move on to the proposal stage. Okay, so if throughout this day you don't know maybe how to write a good abstract, that would be a really good question for the 5-minute, what are they called again? Sidebars. Thank you very much. All right. So make sure that if you're, because really we want you to be successful, right? And kick me out if I'm too long. Okay. So please make sure that you fully understand what an abstract is. Make sure that you understand it as written within the BAA. And I'll get off of that. Right. The evaluation criteria are listed in the presentation. And just to note, the presentation will be provided to you. It will be put on sam.gov, I believe, or at least at a minimum on the ARPA-H website. It'll be in various different places. So these slides will be available to you as well. Make sure that you're submitting your abstract and your proposals to the correct place. And for some of this, really, I'm going to say you need to read the BAA, right? Because what I'm saying today is not the official, like the official language, right? So everything in that BAA, that is your official guide to be able to be successful, right? So whatever I'm saying today is trumped by that BAA. So I will always point you back to that. Let me see here. Again, so the full proposals, right? That is only if you have a successful abstract, right? And the evaluation, so I will note that abstracts are evaluated underneath evaluation criteria 1. And then if the proposal is then, or you're asked to provide a proposal, right, it would be 1 through 4 with descending order of importance. All right, these are the evaluation criteria. I am not going to go over these in detail, but again, I am going to point you back specifically to the BAA. Okay, please get to know these well, because you will want to make sure that they are in your proposed solution.
Are references included in the page?
Are references included in the page limit? No, they are not. Absolutely. And make sure to check the BAA just to make sure. Right. And here's some more. I will say that, as you can see, as I mentioned earlier on in the presentation, that right cost, price, that is the last evaluation factor. Again, that doesn't mean that it's not important. It just means that we're more interested in your technical solution. All right, some final guidance here. I think I've mentioned most of this, but please make sure to monitor sam.gov. It is very important. Monitor ARPA-H website because we put a lot of information on program-specific tabs. And of course, if you have any questions, you can submit them to the FAQs. Hopefully, we can address a lot of your questions here today as well, but they likely will come out in written form just to make sure that we inform the entire public. I think that is it. Any questions before I hand it off to somebody else? Perfect. Back to you, Andy.
Great. Thank you, Jennifer. And so just to go back just real quickly, dates and deadlines. And, you know, I apologies for the quick turnaround. I know if I was on the other side, I'd be cursing the program manager that, you know, built these aggressive deadlines. But the abstract due date is December 1st at 9 a.m. So whatever you do, if you are interested in working on APECx, please get something in by that deadline. We're anticipating quite a few abstracts, and so there will not be exceptions made if you're not able to submit. The submissions are done through ECPS, which is an HHS system. I don't believe our link is live yet for the abstracts, but that will be live soon. And like Jennifer mentioned, that will get posted on sam.gov as well. And so refresh that on a regular basis. You know, from a program management perspective, to keep to these timelines, we're anticipating getting answers on the abstracts back within a week. So December 8th is our deadline to return feedback, both positive and negative, in terms of requesting a full proposal or not within a week. And then the full proposal due date is January 19th. Right. And so that's 6 weeks. And again, there's holidays in there. So not a ton of time, and apologies in advance, but you know, there are reasons why we have these aggressive timelines. So we have an inbox for the team that we all see and monitor every day. It's there on the bottom, so please leverage that. We have the sidebars both in person this afternoon as well as virtual. And so we are happy to talk with anyone and everyone who's in the audience, both virtually and in person today, about questions they might have related to the process. We also are going to have an FAQ session before we go to lunch, in which we'll answer a lot of the questions that we've received so far. Those FAQs are also posted on the website. I'm going to introduce Dominic, who's a part of my team. He's our business and financial manager, and he's really the interface between all things program related on APECx from an administrative perspective and you all. So Dominic, I'm going to stand up here and move your slides. So just let me know when you want to go to the next one.
Right, thank you, Andy. Good morning, everyone. My name is Dominic Brown. Been with ARPA-H since beginning of June, contractor supporting the APECx program. So I'm the business financial manager. So here's start off talking about a little bit of the contracts and how the cost proposals should work. Look at your checklist and the guidelines and the requirements. Moving forward, so here's some of the FAQs that we usually see here. So like Jennifer said, please read the R&D solicitation here and the FAQs prior to submitting your questions. And the questions will be answered via FAQs on sam.gov. So all inquiries shall be sent to the APECx at ARPA-H.gov email address. Any questions that you do have, please make them clear as possible. And one main note here is please do not submit any sensitive information from other companies and or other individuals. ARPA-H may rephrase questions and consolidate some of the questions, and this is just for administrative purposes if needed. And then if you have any conflicts in between the FAQ and the solicitation, always defer to the solicitation, please. Next slide, Andy. So here's the review process for APECx for the abstracts and proposals that you see on your screen. So on the abstract side here, of course, like Jennifer's mentioned earlier, submitted via the ECPS program database, and that will be reviewed upon the submission deadline. So it will not be reviewed before. And so the compliance and responsiveness review. So this is very important to pretty much what we look at. We'll hear if the format requirements, it meets the standards, if it was submitted on time, and if it does, match the technical requirements that we're looking for within the program. So then at that point, we'll pretty much move over to the review assignments that are made. Once they made, we review, and then at that point, the program manager will make a recommendation. And then the last one, the notification will be sent. So you see three items here that you'll either get an invite, not invite, or we'll be looking at some of the requirements, but we may ask for additional information. So if you look on the proposal side, it's pretty much the same standard, same items, but you got a couple that's added here. And those that's added here is this number 8 and number 6. As you see, the SRO reviews recommendation and determination. And then if you get notified, if you select it, we're going to negotiations. Next slide, please. So this is kind of what you heard Jennifer explain about the cooperative agreements and the other transactions. This is pretty much a checklist of the requirements that we kind of are looking for. I'm not going to go line by line here, but some of the main ones you see is the cost proposal here on the Excel standard spreadsheet. And also the sections A through E is 30 pages max. And of course, F through I, you do not have a page limit. And the other one here is the forward price rate agreement. And then as you look over to the other transactions, pretty much it's kind of the same list here. Pretty much has changed here. That's here. We got the volume 1 technical management proposal. And as so, you still got the standard cost spreadsheet through ARPA-H. So here's the cost proposal suggestion and reminder. These are kind of the do's. So you want to provide the requested information on your proposal cover sheet and your proposal submission. So this is to include the cost estimate of the total funds that you request from the ARPA-H and the APECx program, and the amount of any cost share. So per task should be broken down by fiscal year and the phase. And then the summary projected funding requirements by month, all phases of the project. And so the next one here is pretty important. So anything over $10,000, we require a backup quote. We require purchase evidence, a detailed explanation of the labor material costs that's needed. So if you give us a cost proposal and we see that's over $10,000, I'm gonna come back and request evidence of the $10,000 of why you're over 10,000, and we need to submit that as soon as possible. So also with your subcontract, you need to itemize the subcontractor's cost proposal also, and that needs to be at the same level of detail as your prime. So the prime is going to be responsible for the subcontractor's cost proposal. So if you feel like the subcontractor's cost proposal is not in detail to your liking, it may not be to ours. So therefore, you may need to work with them to get it very detailed on that level, to understand that we need that in our hands to get it going. So the last one here is just to make sure we can state this more and more. Just make sure you're realistic about your costs. We understand sometimes that you may underbid or you may give us a low cost just to try to get the proposal and the negotiation get selected. Please do not do that. Be as real as possible. Be as transparent as possible. And then we'll be needed to successfully accomplish the technical approach. Next slide.
Hey, Dominic, can I add something just real quick? So this is for a full proposal submission. And so for the abstracts, rough order of magnitude budget, we've gotten some questions if we can provide the cost proposal template ahead of the full submissions, and we're going to do that. And so that will post on the website in sam.gov either today or Monday, as I understand it. So it will give you the template to, again, estimate budgets that are going to be required to do the work. And that's what we're looking for in the abstract phase. These are suggestions and reminders that are important to keep in mind for the abstract, right? But that are required for the full proposal submission.
Right, and so of course you had your do's there, and here's the don'ts. Don't forget to utilize the cost proposal checklist, as Andy was stating. And one thing we do ask, do not change the formulas in the spreadsheet. If you do have to, make sure the total amount tab does accurately reflect the cost corrections. I emphasize that again, please do not change it unless you totally have to. And next one, of course, please do not delete a row or column if it does not apply to you. Just ignore it, move on, go to the next line item. If it does, it may increase the time of processing and looking over the cost proposals. And so once again, don't strategize, don't cost strategize. Don't just be realistic as possible with your costs. All right, so as we went through the do's and don'ts, here's the checklist. So like I said, this will be presented to everyone after this. So pretty much the main one that we went over is don't go over the 10,000 line 6. Please have your evidence there, a detailed explanation of why you're over 10,000. It's just to give you an example, if you got 8 technical seaters that needs to go on a monthly battle rhythm meeting, does all 8 need to go? And we need a justification if all 8 needs to go. So that there, number 11 is that your proposal include response regarding the organizational conflicts of interest. And then not the last one, does your proposal include a complete data rights assertion and table certification? And that is it from me.
Okay, Dominic, thank you so much. And I know there was a lot of information there. A lot of that's included in the solicitation that's on SAM.gov on the website. We'll also be posting all of these, right? And again, we're happy to take any questions, as you come across them in the abstract formulation phase. We're actually on time, which is fantastic. So before we go and caffeinate, we've got Dr. Jenica Patterson, who's going to talk about our Project Accelerator Transition Innovation Office. And, at ARPA-H, we are very interested in the basic discovery elements and the novel technologies and things that are going to come out of our programs. But we're also there to partner right all the way through that development lifecycle. And we really want the innovations and the discoveries we make to translate into things that people can use. And so Jenica helps manage an entire office that's focused on providing those capabilities to performers so that the things that we do end up making and discovering and creating, right, end up living in the wild after APECx is over.
Thanks, Andy. I'm so excited to be here this morning. So I want to say, I know I'm here standing between you and a break, but I want to say it's going to be worth your while. And the reason is because, number one, if you are awarded under Andy's program or any other program at ARPA-H, you will work with our office. Number two, regardless if you get an award at ARPA-H, you can still work with ARPA-H through some of the services that we provide. So I'll tell you a little bit about that. So I am one of the division directors within the Patio office, which is our transition office. And we have 3 divisions within that office. The reason why this office is so unique, especially with other government agencies and including other ARPAs, is because our transition partner is really complex. It is every single American. That is 300 million people plus. So that, number one, is complex, as well as we are the health agency. So we're creating technologies that need to jump through many hurdles, regulatory hurdles, reimbursement hurdles. That's going to be really challenging. So because of that, Renee and others decided to create an entire technical office around that. And that's where Patio comes in. So what is Patio's mission? So we're really focused on increasing the probability that the solutions that you might be creating for ARPA-H will, what Andy has called, survive in the wild. So what we do within the office is we provide transition and commercialization services to make sure we can help you work through those barriers. As Andy and I think Amy mentioned, we do not fund performers in perpetuity. Once the program is done, it is done. However, we're here and working closely with the performer teams and with the program managers to make sure we're going to increase those odds for you guys to be successful. So we hopefully can help, for example, attract private investment or really get customers to translate those breakthroughs out into the hands of all people. So how Patio was created was through something called the IDEO framework. So we really modeled our office on that. It's a combination of feasibility, desirability, and viability. And with all of those three combined, we're hoping that we can really create those breakthrough innovations that we can translate to everyone. So for feasibility, that's really focused on the technology. So that's what Andy's team has helped develop, as well as you all here as potential performers, really focus on that feasibility to de-risk the technology. Really, the secret sauce of Patio are the other two things. So that is the desirability and the viability. So desirability is really creating technologies that people want to enthusiastically adopt. And I'll talk a little bit more about that. And viability is really focusing on technologies that the market want to enthusiastically adopt. And we're hoping to bring in those services and support to help with our ARPA-H funded solutions. So you might be asking yourself when you're creating your proposals, where may my technology go? And so these are 7 different areas where we think a technology might translate out into the hands of all people or to the wild. So I encourage you, while you're developing your proposals for Andy's program, to really think about this. Think about what are the barriers that you're going to have to go through to be able to maybe create a startup or go into a nonprofit or integrate into a healthcare system and be able to understand what those barriers are, because when you come to us, we can help and support you in that. So everyone talks about transition at the end, and they say, well, we're going to swoop in and come and support you at the end of the program when the technology is created. And that's not true. Patio starts from the beginning. We've supported Andy and others to be able to make sure that they are building in transition metrics into their programs during program design, during their solicitation development, and on. So we're really excited that we can be a part of every single step of a program. We're right now in between solicitation development and early program performance. And actually, we're really excited. We have an expert that we brought in through our Patio office here to talk about equity. Amanda, she's in the audience. You can raise your hand. There she is. She'll be talking after the break about equity and what she has helped Andy and our team do. So like I said before, Patio has 3 divisions. One of the divisions is mine, that is the Tech Transfer and Transition Division. And one of the services that we provide for our program managers and potentially our performer teams is we have something called Expert in Residence program. So that's really bringing in that private sector expertise to really increase, remember what I said, the viability of technologies. So we want to make sure that we're embedding experts with our program managers and our performer teams to really increase the chance that those technologies will go into the market. So we can do things like understand what are the regulatory hurdles you have to go to. Can we bring a reimbursement expert into your team to help support? We have regulatory reimbursement experts, commercial focused experts in residence. And again, we have Amanda here as one of our equity focused experts in residence. Another one of our divisions in Patio is the Health Ecosystem Engagement Team. So really what they do is help with desirability of what we're creating at ARPA-H. So Amy presented that circle of in the beginning about stakeholders and communities. That's really where this ecosystem engagement team helps swoop in. So they're really accessing ARPA-H with resources and expertise of the health ecosystem through what we call a network on demand. So we're going to leverage the institutional knowledge of everyone out in the nation to be able to bring that back for us to make better programs. We were here earlier in the week meeting with people, some stakeholders, some communities, some universities in Colorado, just to understand what you can bring to bear potentially for performers or for program managers in the future. And the last division we have is called the ARPA-Net for Health. You might have heard about it. It's gotten a lot of buzz from ARPA-H. And it's really a great opportunity for us to help meet people where they are, to meet every single one of those Americans. So it's funny because Congress actually mandated that we had 3 geographical sites across the country. And so we took that as an opportunity to have 3 hubs located across the country and created an opportunity to have those hubs have spokes that can connect to the entire nation. So every single state can be connected with ARPA-H. And that's really an opportunity, again, to meet people where they are. So we have created 3 hubs. 2 we actually solicited out to the world, and one is located in our DC area. So that's our stakeholder and operations hub. That's where a lot of us are located, an opportunity for us to meet with federal stakeholders. It's really important for us to continue operating. But with our competitive site selection process, we reached out to the nation and we're like, who wants to work with us? So we had an announcement come out where we identified 2 other hubs across the country. And that is the customer experience hub. So remember when I talked about desirability, that's bringing in technologies we want for people to enthusiastically adopt. That's the whole purpose of the customer experience hub. And then that is located in Dallas, Texas. And we have a contract management firm that supports that particular hub. It's called ATI. And they're really going to be able to activate the network for us and bring in these services that we can support performer teams. And then the other one is the Investor Catalyst Hub. So that's bringing in, again, that viability. So we actually have the hub located in the greater Boston area, and it is run by VentureWell. And many of you know, if you work at NIH, might know VentureWell. They actually help support the RADx initiative. So I talked a lot about the hub, right? So the hub is really that physical location, that nerve center of the network that's located in 3 distinct locations. Really where we think the muscle is for our network is the spokes. So the spokes can be communities, it can be industry partners, patients, hospitals, community groups, academic institutions. And really what we can do is convene that network across the nation to be able to support our performer teams and our ARPA-H programs to really meet people where they are in America. And you're probably asking yourself, well, who is a spoke? Who can I become a spoke? Absolutely. Regardless if you are a performer team or not, you can go to the ARPA-H website right now. And if you think you bring that unique capability for ARPA-H, either in customer experience or Investor Catalyst Hub, we encourage you to go and submit your application and become a spoke. So I'm just going to talk a little bit more about what the Customer Experience Hub could do for Andy's program. So again, the Customer Experience Hub is really focused on the human-centered approach. We want to create technologies that people want to enthusiastically adopt. Andy mentioned there's a lot of vaccine hesitancy in our nation. So what if we have the opportunity to do a network activation call within the entire nation to bring the voice of the end user and the voice of the customer to understand why that is? What would make them want to actually use Andy's technology? How can we create a technology that is designed that people are more comfortable using? So that's just one example of how we could use the Customer Experience Hub. And lastly, again, our Investor Catalyst Hub. So Andy talked a lot at the end. He really wants these technologies to survive in the wild. That's really what the Investor Catalyst Hub can do. It can help support you in your regulatory or legal needs, intellectual property. What if you don't know who a GMP manufacturing groups are across the nation? We could do a network activation call within the Investor Catalyst Hub to be able to support you if you become a performer. So we really want to make sure that this Investor Catalyst Hub is helping you bring your product out into the market. So that, I think that's all my slides. I just want to say thank you for listening. I hope you now understand how we can really support Andy and his program and potentially support you as a performer team. And regardless, I really encourage you to, if you think you have that unique capability, join our network, send in that application and be part of the ARPA Net for Health. All right, thank you.
Okay, so yeah, I want to foot stomp something Jenica just mentioned. I'm not going to own the IP. I'm not going to develop these candidates, right? You guys will. And so like these services are being brought on board within our agency to really be tools for you to use. So definitely find Jenica throughout the day and ask her some questions. An example, we have an SBIR performer, right, who's developing some really cool technologies, but they haven't really applied it in a clinical setting before. So we've started some entrepreneur expert in residence discussions with them, right, to bring in someone who's familiar with developing medical device products and doing clinical evaluation, right, and we're pairing those people together. So those are some of the things, right, that as a performer you'll have access to. Also good to consider in the abstract and full submission phases, right? We are very, we're pushing on the translational aspects, commercialization and things like that as part of the APECx program. We don't expect everyone to have a formal legal agreement with a transition partner from the beginning, right? And so definitely take a look at the services that Patio and others can provide. And those are potential additions that can be brought into a full proposal, right? To really fit and fill some of the metrics that we have defined for APECx. Okay, so we have a break. We're reconvening at 10:25. In terms of coffee, there's a coffee shop downstairs in the lobby, and there's a few places right outside. And I would ask everyone to be back at 10:25 when we're going to go into our government enabler capability discussions. And for any of the folks who are presenting, if you could just pop up here real quick before we go on break, just so we're all on the same page. But that is, again, it's another opportunity, just like the Patio services with these government enablers, to build in things to your program, right, that might not be accessible just out in academia and industry. And so we're excited to hear from our government partners here in a few minutes. Thank you. Okay, we are going to go ahead and get started. So everyone, please come in and take a seat. And for those of you who are virtual, we are going to roll into our next session, which is focused on our government capability enablers, right, and the partnerships that we're building across the U.S. government, right, to help with the objectives of APECx and for performers who are going to be working with us. I believe Kim Taylor from NIAID is going to be our first government presenter. And so, Kim, are you able to hear us?
Yes, I am. Can you hear me?
Yes, we can. Great. So John here is going to help run the slides as well. And so if you just want to let us know when to move forward, we will get after it. Just for all of our presenters. So Kim, you have 15 minutes. All of our national lab partners, you have 10 minutes. And then Danny at the end of the session has 15 minutes. And so we will be very abrupt with time because we've got a lot to get through. So appreciate the cooperation.
Good morning, everyone in Denver. Sorry, I'm not here or there with you today. My name is Kim Taylor, and I'm a senior scientific officer with the Concept Acceleration Program for Vaccines. And I am from the Division of Microbiology and Infectious Disease. And we are actually an extramural division at NIH, which means that we support basic and translational research and all the way through the clinic, typically phase 1, phase 2, and product development, mainly through grants and contracts within our extramural division. And later, there'll be someone from the VRC talking from the intramural perspective. I was asked to talk to you today about some of NIAID's collaboration and funding opportunities that can be leveraged for AI, antigen design, and product development. And after hearing Andy's talk, I think, it's going to the APECx program is really going to complement what we're doing quite well. Let's go to the next slide, please. All right. So as many of you know, during the peak of the outbreak or the pandemic, COVID-19 pandemic, the G7 met and put forward the idea of the 100-day mission. And this is really to respond to future pandemic threats. And the purpose was to transform our capabilities to really meet this extremely expedited timeline. The USG also came out with AP3, or the American Pandemic Preparedness Plan, to address what capabilities we need to meet this 100 days. Next slide. And as such, NIAID also put out our pandemic preparedness plan and in December of 2021. And this is really in direct alignment with the AP3 and describes two major approaches for vaccine development to address emerging infectious diseases. And so the first is really the priority pathogen approach, where products are developed specifically for high priority pathogen targets, such as anthrax and filovirus. You develop it all the way through licensure and then stockpile. Next slide, please. But the second is the prototype pathogen approach, where strategies or capabilities are really developed and they're built on prior experience. And SARS-CoV-2 is a great example of this, where previous experience coming from the VRC, from MERS and SARS and vaccine platform development quickly led to a solution and EUA of vaccines in about a year's time. So next slide, please. So pandemic preparedness, it's really all about being prepared and ready to respond. So this is when solutions are pre-identified and that we have them, that they're likely going to work for unknown and newly emerging threats. This includes knowing what vaccine antigen design, what platform, what are the correlates or the mechanism of protection. That's for vaccines, and for monoclonal antibodies, it's what are the epitopes? What are the functional properties that we need to have good candidates and products? And so there's also a need for rapid manufacturing and deployable technology platforms that are going to be needed for newly emerging threats. Next slide. So really, I think the AI will play a huge role in really advancing some of this. As we've seen before, machine learning will be key, and many of these computational methods, such as structural biology, et cetera, are really going to advance our understanding and accelerate development. Next slide. So as such, to address pandemic preparedness, NIAID put out a new initiative in 2023 called REVAMP, and that's research for vaccines and monoclonal antibodies for pandemic preparedness. And we anticipate the award sometime in the spring of 2024. So what the REVAMP centers will do, they're going to be very collaborative and conduct multidisciplinary research on prototype viruses from a select viral families of pandemic concern, and those are shown on the right. This will include basic and translational research through IND-enabling research on prototype pathogens, and develop vaccine and monoclonal antibody strategies that will be able to apply to other viruses within that viral family. Next slide. Here's another look at the REVAMP centers and what they will do. We're going to have multiple centers that will form a network, and together they're going to be conducting research on known prototypes, and through proteomics, epitope mapping, learning more about the viruses, they're going to have build foundational knowledge, viral structure, protein structure, which is really going to be the key to develop generalizable vaccine and monoclonal antibody strategies. Next slide. We envision that these centers will really leverage computational methods and AI tools throughout the whole program. And by doing so, we also have a coordination center and data sharing center that will pull together this knowledge and be able to share it with the network, and also collaborate beyond the network. So I think this will be a huge opportunity to become involved in. Having this network up and running and interacting with key stakeholders across the globe will really stimulate research and collaboration, including, I think, the APECx network that you all are developing. Next slide, please. Okay, so I wanted to point out a couple of our resources that we have that's been working really well to help the scientific community. And the first is a structural biology centers called Crystal ID. And the efforts here are focused on targeted pathogens causing infectious disease. There are 2 centers for this, and through this program, NIAID is able to provide the scientific community with 3D atomic structures of protein, expression clones, purified proteins, functional characterization of really select, very important targets, and structure-guided vaccine and drug discovery efforts. Next slide. Now, I wanted to provide a couple examples of what these centers have done in the past, and I think there are a couple of really great examples here. The structural biology centers have really advanced technology platforms for both vaccines and therapeutics using using computational modeling. University of Washington, as part of one of the centers of Seattle's Children's, developed the world's first approved nanoparticle vaccine that was subsequently EUA'd in Korea and then pushed out also by the WHO, where UW designed the self-assembling protein nanoparticle using the Rosetta Fold software. And so this nanoparticle, which displays 60 RDBs, was shown to be highly immunogenic, and broadly protective vaccines are also now being developed using the same type of nanoparticle platform. Another example is for therapeutics. One of the centers developed a computationally designed what's called mini binder or proteins. And so these mini binders directly compete for the RDB binding site and have demonstrated protection against SARS-CoV-2 infection in small animal models. And these mini binders could be really useful for either as a prophylactic or therapeutic treatments. Next slide. Another program I'd like to tell you about is our PO1 grant centers, which are developing broadly protective coronavirus vaccines. And here is another group, one of the centers led by Bart Haynes at Duke University, and also in collaboration with Betty Korber at Los Alamos National Lab, are really using these AI and computational strategies to track evolution of SARS-CoV-2, taking spike and other antigen targets, and then design immunogens to cover multiple variants and also more distantly related coronaviruses. They're also using a variety of vaccine platforms. Next slide, please. I also wanted to switch gears a little bit here and talk about funding opportunities that don't directly have AI, but where AI can be certainly leveraged and utilized moving forward. And so the first is our Omnibus Broad Agency announcement, or it's a product development contract where we provide direct to investigator funding through phase 1. It has capabilities for development of vaccines, therapeutics, diagnostics, and really the focus is on technology platforms. So new vaccine platforms, new adjuvants, alternative delivery systems. We also like to see delivery of products through mucosal routes or oral routes, and also broadly protective vaccines. I'd like to point out that this is focused on our high priority pathogens, and they do include the RNA viruses of pandemic potential as part of our broader list. And I suspect that this might be a great area to really leverage AI to develop future vaccine candidates. Make sure you keep your eyes peeled for this initiative, as it typically comes out in the beginning of the year in January, and then applications are due in April. Next slide, please. Another opportunity is our preclinical services and research resources, where these are leveraged to address key product development gaps. And the goal is to de-risk product development and technologies and to help advance candidate products. I want to point out that the capabilities don't include AI. However, again, that if you are developing a vaccine candidate through computational methods, PCS could be leveraged to evaluate your vaccine candidates in our animal models. And also taking that data generated through PCS could be leveraged for further machine learning. Next slide, please. And this is particularly useful for outbreak response and trying to rapidly assess and evaluate interventions. Next slide. One more last kind of heavy slide. I wanted to point out that our other division, DATE, has multiple databases and computational tools that cover adjuvants, where you can look for various partnerships, and you can pull out data that could actually be interfaced perhaps with your own AI that you will be developing. And so next slide, please. I'm just going to wrap up here and point out that you can sign up for email alerts for further funding opportunities, and I've listed several opportunities that may be interest of you as a performer. Thank you very much for your time, and feel free to contact me if you have any other questions regarding our programs.
All right, thank you, Kim. Our next speaker is Harshini from Lawrence Berkeley National Lab. Harshini, are you able to see the slides and present?
Yes, I'm able to see the slides. Can you all hear me?
Yes, we can hear you.
Great. Thank you again for the opportunity to talk about some of our capabilities at Berkeley Lab. And I'm very sorry that I'm not able to be there in person today to meet all of you. Next slide, please. So the scale of ARPA-H programs actually ranges a whole variety of different targets, which clearly requires multidisciplinary science with an attention to security, whether it's biosecurity or cybersecurity in the case of data, evaluate and ensure translational value of the outcomes, and provide unbiased evaluations of whether things are able to translate to the real world. And hopefully capitalize on core resources with some degree of agility in order to transition things from foundational science to usable solutions. Next slide, please. In a nutshell, this actually is the scope of how the Department of Energy National Labs also operate, looking from science to technology and developing multi-purpose capabilities that can span the entire realm of science that clearly can feed into these programs at different levels. You'll hear from other national labs today as well, but at Berkeley Lab, we are on the science end of the spectrum. And I'm going to take this opportunity to give you a little bit of an overview on Berkeley Lab and the bioscience capabilities here, while delving into a little bit of more detail about structural biology capabilities, which is the most relevant to the APECx program. Next slide, please. So Berkeley Lab is one of the premier institutions in the world, and it was the first of the 17 national laboratories, although it can be argued that Los Alamos, my former alma mater, was the first as well. So I'm not going to arbitrate that point. But it has major user facilities, which are clearly capabilities that are available to programs such as ARPA-H programs to enable cross-sectional deep foundational science in different areas, which are not easy to build from scratch up for every single program. And that is the whole purpose of these government resources, is to be able to be available and enable science in a broad swath of scientific realms. Next slide, please. These user capabilities include the Advanced Light Source, which we'll talk about in some detail, Supercomputing Center, and perimeter, the supercomputer is shown over here. We have the Joint Genome Institute in the middle, which is the sequencing hub for the Department of Energy, the Molecular Foundry for manufacturing, and ESNet, which is the Energy Securities Network Center. We also have NERSEC and several large project centers at Berkeley Lab, which enable bioscience and related innovation and development. Next slide, please. Bioscience at Berkeley Lab is pretty large. It is arguably the largest bioscience capability within the Department of Energy National Lab Systems, with almost 1,000 individuals, several scientists, postdocs, faculties, and others that are available. We have 3 scientific divisions: the Biological Systems and Engineering Division, Molecular Biology and Biophysics, Molecular Biophysics and Integrated Bioimaging Division, and the Environmental Genomics and Systems Biology Division. We also have the Joint Genome Institute, which is a user facility within biosciences. Next slide, please. So JGI offers a lot of capabilities in sequencing, including databases and how to integrate disparate data, metabolomics and proteomics with sequencing data, archiving the data, availability of data in a usable format for different people. It also does sequence facilities, and I think this is a useful capability for a broad swath of biological science programs. Next slide, please. The Environmental Genomics and Systems Biology Division is more focused on testbeds and evaluating capabilities. And although these programs were developed with the context of environmental microbiology, the testbeds have proven to be extremely relevant as controlled ecosystems for a variety of health different applications as well. Next slide, please. The molecular biology and biophysics and integrated bioimaging division is perhaps the most relevant to this call, and I'll spend a little bit of time talking about it. This division, led by Junko Yano, shown here, studies the fundamentals of biology, and it actually is science at the interface of biology, physics, chemistry, computation, and nanoscience. The goal is to actually achieve measurements across length scales and time scales in order to characterize, evaluate biological systems, but also resulting in multimodal imaging and structural biology capabilities that it can advance the state of the art when it comes to characterization of new targets in a variety of environments. This work has actually led to the development of software like the Phoenix platform that has been used widely and effectively by thousands of people and companies across the world. It also has led to several drugs and products that have transitioned from the characterization to the use. A small subset of probably the most popular one is shown on the right that includes drugs like Herceptin, which is used for the treatment of breast cancer. Next slide, please. This division involves the researchers that work with the advanced light source in order to do biological characterization and structural biology. That's the focus for our participation in the APECx program. And so I'm going to spend a little bit of time talking about these capabilities. So when you're actually looking at characterization of a target, you really do not know what method is the best method to bring forward. And usually we are biased by the availability of specific set of resources that we can actually bring forward. Somebody might have an X-ray crystallography, somebody might do small angle scattering, but the plug and play and the mix of these technologies is actually really critical in enabling an agile response, as was evidenced during COVID-19, which I'll talk about in the next couple of slides. To this end, the advanced light source actually offers capabilities in macromolecular X-ray crystallography. It's supported by Howard Hughes industry universities, as well as largely by DOE, BR, and NIH, which is an HHS program as well. We have soft X-ray tomography, which is a unique capability, which is present only at ALS. And that actually allows for rapid three-dimensional reconstruction of biological samples, even in a complex background without purified antigens and crystals being available. We have cryo-electron microscopy, which is an NIH facility. We have small angle X-ray scattering, which is extremely useful for rapid characterization in high throughput. And we'll talk a little bit about that in the next couple of slides. X-ray footprinting, which probes interactions between proteins and can give you very valuable information on binding energy. Spectroscopic capabilities, including infrared and multimodal hyperspectral imaging for life samples, diagnostics, metabolites, as well as an integrated unit that is dedicated to data analytics, software, application of AI and ML for structural biology within this division. Next slide, please. I'll be skimming through this, but happy to talk about it in greater detail if anybody is interested. But essentially, the way that I like to explain this is that some imaging technologies are really good at taking a picture when something is sitting still and has high resolution, and you have a beautiful camera, the background is clear, like I showed with the first bird on the left. Others are capable of doing the same, but if the background is a little murky, they can still do a decent job. The third set is, what if the bird is flying, it's constantly in motion, and the background is also noisy. So you need tools to be able to do all three of these. X-ray crystallography is an example of the first, cryo-EM is an example of the second, small angle X-ray scattering is an example of the third, for instance. We've been able to apply these tools in different combinations, especially during COVID-19. And I would arguably say that most of the antibodies that we actually got out, the structure was resolved at the ALS. This is sotrovimab, which was from GlaxoSmithKline and Vir Biotechnology, was characterized at the ALS, followed by some work that I show on molecular mechanisms of interactions of drugs with viral envelopes for SARS-CoV-2. Understanding and unraveling the RNA copying assembly of SARS-CoV-2. And then obviously beyond infectious disease, I just threw leukemia drug from Gilead that was actually developed using a combination of these methods. Next slide, please. So a lot of this actually comes with a backbone of data science to enable discovery. So there's multi-scale, multimodal visualization techniques so you can understand this awareness. And I show a couple of examples of that over here. During the COVID pandemic alone, over 120 atomic resolution crystal structures were generated at the ALS. We did solution state analysis. We did observation of real-time infectability to the receptors. There was visualization of novel therapeutics, as I showed, as well as the viral protein interactions using a combination of different methods. And all of this data was also integrated and refined via AI ML mediated backbone capabilities that have been developed, but also integrated in a multimodal fashion so that you can look at combinations of data streams and the value that they bring for your structural resolution and not one particular method alone. Next slide, please. I'm not going to belabor the rest of the slides here, but I did throw several slides to talk about other capabilities at Berkeley Labs, such as biological engineering over here. Next slide, please. And the use of spectroscopy as an agnostic diagnostic, again, within the ALS, which is available for performance to use if required. Next slide, please. And finally, I want to just talk about biomanufacturing because a lot of the things that we are talking about here may require scale-up science. And the Department of Energy has invested a lot in terms of actually understanding how to make different things, how to have automation, self-driving labs to sort of enable manufacture in an American economical sense, as well as scale them up, as I show via 3 major projects here, the Joint Bioenergy Institute, Agile Biofoundry, and the Advanced Biofuels and Bioproducts Process Development Unit. Whereas all of these were developed for industrial biotechnology efforts, we actually have clear and concise examples of them being used for health products, including the manufacture of adjuvants, for example, and certain drugs, and scaling them up in a fermentation system, which is really a value added in terms of thinking about translation of some of the drugs and products that ARPA-H may be producing. I do have additional slides which I can make available for your review on additional capabilities at Berkeley Lab, but I think I'm almost at the end of time. So I'd like to stop here and take any questions on the chat or later based on how the organizers prefer. Thank you so much for the opportunity to discuss some of our capabilities with you.
All right, thank you very much. Our next speaker is Anush from Oak Ridge National Lab. And are you able to see the slides and present?
I can see the slides. Can you confirm that you can hear me?
Yes, you are good. Thank you.
Fantastic. Thank you. Very nice to meet you all. My name is Anuj Kapadia. I'm a scientist at Oak Ridge National Lab. I lead a section on advanced computing and health sciences. My apologies as well in not being there in person. I was supposed to be there today. Unfortunately, I had a health emergency that precluded me from traveling. So doing the best that we can online. Next slide, please. For those of you who don't know Oak Ridge National Lab, this is us. We are in the mountains, in the Smoky, close to the Smoky Mountains in Oak Ridge, Tennessee. It's about 20 minutes from Knoxville and about 3 hours east of Nashville. Oak Ridge National Lab is currently home to the world's #1 and #5 fastest supercomputers ever. Next slide. For many years, Oak Ridge National Lab has had core missions in a variety of subjects ranging from neutron sciences, computing at scale, materials and chemical processes. We have additive manufacturing. We have a manufacturing and demonstration facility for prototyping technologies. We've had a strong footprint in fusion and fission, energy production, and just technology development in general. Biological environmental systems, isotope productions, all of these that you see on the screen represent our core directorates, if you will. Next slide. A big part of this, and what I'm going to talk here about of interest to APECx, is the Oak Ridge Leadership Computing Facility, or OLCF. Next slide. OLCF has had a long history of providing the world with some of its fastest and most capable computing systems, ranging from Jaguar in 2009, followed by Titan, then more recently Summit, and currently Frontier. Each of these systems has represented about a 10-fold increase in computing capability. Frontier, which is our current flagship system, is the world's first exascale computing system. That means 10 to the 18th flops, floating point operations per second. It's roughly equivalent to 50 million laptops. Next slide. This system was launched last year. Yeah, I think it was launched in 2021 and then benchmarked last year in 22. At the time of benchmarking, its performance was 1.194 exaflops. The system itself contains 74 cabinets with over 9,000 AMD CPUs and upwards of 37,000 GPUs. We have 700 petabytes of storage. It's also an extremely economical system. It was ranked number 2 by the Green 500 in terms of energy usage. Next slide. This system is not something that was built solely for Oak Ridge National Lab. It's part of DOE's user facility, which means collaboration is a big part of what we do. We already collaborate with over 1,500 users. There are 240 active projects. A large part of Frontier is devoted to doing collaborative science with the user community, with you, essentially. We have collaborated with multiple entities. There have been several Gordon Bell finalists and several Gordon Bell prizes. For those who are not familiar, Gordon Bell is a prize that's established to identify projects that have pushed the limits of supercomputing capabilities. Much of Frontier is in a user program. Right now it's called Summit Plus, which should be accepting proposals, or the deadline may have just ended. In general, it is available to users such as yourselves. Next slide. So how do we partner with ARPA-H in bringing computing capabilities for the benefit of viral antigens? We have several mechanisms in place for collaborating with you all. The way ORNL, the way Oak Ridge National Labs performs these collaborations is through strategic partnerships. We can do subcontracts. We have user programs. In fact, an interagency agreement was just established between ARPA-H and Oak Ridge National Lab that would allow us to work with any of the ARPA-H performers. So anyone who is interested in exploring projects in using ORNL's high-performance computing facilities or its people, please reach out to me. My name and email address are both on the screen. I will be your point of contact, and I can help put you in touch with the right groups from the computing perspective, or anyone from the biosciences, or things related to protein discovery and topics like that. Next slide. A big part of who we are and what we do is not only the facilities, but also the people. Yes, we have fantastic computers, some of the best in the world. A big part of this is also the people who know how to use these machines to explore and achieve certain targets that would not otherwise be possible. We have embarked on several projects in medical discovery and predicting protein structures. We have been looking at quantum NLP, natural language processing for medical text. There have been projects on cancer detection, cancer surveillance, and then a considerable amount of work on SARS-CoV-2 reproduction as well. All of these represent core capabilities at Oak Ridge National Lab, all of which focuses around the computational aspects. Next slide. So I think that's all I have. I am happy to take questions or answer questions in the chat, whichever is best.
All right, thank you very much. We're now going to move on to our in-person speakers. And our first speaker is Josh Adkins from Pacific Northwest National Lab.
Great, thank you very much. So first, thanks to the previous 2 National Lab presenters. I think they give you a sense of National Labs being collaborative in nature and often technology and science focused. So in a space like this, I think there's a lot of value in groups that are able to adapt to the needs of our sponsors. And so that's an important point I just want to make. PNL has a strong science mission. It's across many different activities, including the biological system sciences. the Earth System Sciences. There are user facilities such as the Environmental Molecular Sciences Laboratory, where many of the resources I'll be talking about are available via proposals. Then there's the Chemical Material Sciences and Advanced Computing and Particle Physics. So these are not actually isolated pieces of science. They're pieces where we can pull different threads to solve problems for different sponsors. And so advanced computing, it's pretty clear. A lot of the things that are going on in this particular call are around advanced computing, chemical and material sciences. There's really excellent connections, and then the biological system sciences at the lab are pretty impressive. So for the most part, PNNL is known for a lot of things in biological sciences, but one of the best areas we're known for is in our mass spectrometry area. We do a lot of development of mass spectrometry technologies, both for proteomics, metabolomics, lipidomics. And when we talk about proteomics, this isn't a single technology. It's not about a single measurement mode. We're measuring parts of molecules. We're measuring the post-translational modifications. We're measuring the structure, many of these different things. So we don't have one hammer looking at one nail. We're actually, when we partner, we come and work with people and figure out what's the question they're trying to ask and which tools in the toolbox can we use. Here's an example of a recent study where we were looking at 7 viruses with 11 strains, looking at the impact of those viruses on cell growth, division, and immunogenicity. Never mind. Didn't sleep very well last night. So then some examples of some of the advanced proteomics methodologies. So in this particular case, on the upper, what would be your left is an example of a technology where we're trying to make mass spectrometry faster. This is known as SLIM. And in these cases, we're trying to get away from methods using liquid chromatography. So if you've worked with people doing proteomics, it takes hours sometimes to get a proteomics measurement or a metabolomics measurement. In this case, we're trying to move away from liquid chromatography into gas phase. All this technology is being developed at PNNL, and hopefully can be something that could be transformational in future areas for characterizing and monitoring biological systems. Down here in the lower portion, you can see the little nanowells next to a quarter. This is where we're able to now do single cell proteomics methodologies and to characterize very, very small samples. We use this to help support imaging capabilities, as well as trying to understand cell populations. On the side here, this is one of our very large instruments supported by the Department of Energy, known as Fourier transform ion cyclotron resonance. And these allow us to look at very large proteins in their intact form, so up to 150 kilodaltons. So if you're looking at a protein and you want to understand, well, we knew what the genome said, but what is actually going on with this? What are the ranges of variety that this protein actually exists as, with post-translational modifications and different cleavage states? So another area that I think is of value to this particular group is we have an ongoing internal initiative, which is a fairly large investment by the laboratory to develop new technologies. This one's known as M/Q, which is mass over charge. And what we're trying to do here is, most of the time when you're looking at metabolites or small molecules, in order to understand what it is, you almost already have to know what it is. So you have to have a reference and you have to run your instrument in exactly the same way. It's not very conducive to finding new molecules that are important or of value to your group. So what we've been doing is developing methods that use machine learning capabilities and computation with that ion mobility technology I talked about, to use those pieces of information plus fragmentation to make identifications and quantification measurements without the use of references. At the lab, we've also been working with nanobodies to develop libraries to detect antigens. These are library-based characterizations. We do this in a
cell-free way. We can then pull down, use our other analytical methodologies to determine which is the right binding nanobody to the antigen of interest. We also have a fairly large cryo-EM capability that helps support a lot of the protein work. When we go through and we identify something really interesting, well, now we want to know this post-translational modifications modified this protein. Now, how does it actually interact and what is the structure of that? And we do this in collaboration with our partners at Oregon Health Sciences University. We have a long-running joint institute with them that helps give us access into a clinical teaching hospital. And then their broad capabilities in computational chemistry, AI to accelerate therapeutic interventions and strategies. So you can imagine as we start to know more about the molecules that we're actually getting, not just the sequence from the genes, but actually the post-translational modifications, the processing, we need to be able to do computational analysis of those. So in summary, we have a very high throughput functional analysis for biomarker and therapeutic discovery. We use and develop advanced proteomics technologies for antigen discovery. The nanobody libraries, we do protein structural modeling and imaging, and computational chemistry and AI to accelerate those therapeutic capabilities. So we are in Washington State, but we're in the southeast corner. If you're ever in the area, we've got great wine, look me up, even outside of this program. And I just want to thank you. My contact information is here in the slides. Thank you.
All right, thank you. Our next speaker is Dan from Oregon National Lab.
Okay. So I'll be, I guess I'm batting cleanup for DOE. So another Department of Energy National Lab. So Argonne, I have a very short slide deck. A quick background for Argonne. It was actually the slides they gave me said we were the first national lab. So I don't know what's going on, but so it's outside of Chicago, about 30 miles outside of Chicago, and we have roughly about 4,000 employees. We have a lot of just like the other labs, very multidisciplinary as an Office of Science lab. So we have a lot of capabilities, a lot of world-class user facilities. But for today, I'm just going to talk about 3 of those that are that we believe are directly relevant to the APECx program. Our high throughput structural biology core or pipeline, scalable data management solutions, and then our high performance computing. Okay, so with regards to our high throughput structural biology, the 2 main facilities that support this core are the advanced protein characterization facility on the left. That's where most of the preparation for the proteins occurs. And then on the right, you see the advanced photon source. So within the advanced protein characterization facility, this is where the production, characterization, and crystallization occurs. And researchers within that facility look at the structure and function of proteins. They characterize protein functions and study their interactions with other macromolecules and ligands. So this is where the vast majority of like the wet lab work takes place. Within the Advanced Protein Characterization Facility, we house a couple of centers, the Midwest Center for Structural Genomics. This is funded out of NIH. And this really, it looks to the goal is for rapid determination of structures that are biomedically important, and with the ultimate goal of providing structural coverage of major protein families with sufficient granularity to allow for 3D homology modeling of all proteins using only computational methods. So to provide the base data to allow for direct computational determinations. The Center for Structural Biology of Infectious Disease, similar to the Midwest Center, but solely focused on infectious diseases that have been identified as NIAID as organisms of high concern. And finally, we support the BRAVE program. That's a DOE program. biopreparedness research virtual environment. And with that program, they support fundamental research supporting biopreparedness and response to future pandemics. The APS on the right, Advanced Photon Source, that is it's a synchrotron light source. It produces high energy, high brightness X-ray beams. And this is where most of the work is done. There's a lot of hutches. So we call them hutches. We have 60 beam lines, we call them. So you can see around, you see these little triangles around the ring, which is about 2/3 of a mile around. And each one of those is a separate beam that has been modified for individual uses. So we have roughly, I think, 5,000 or so researchers come to Oregon every year using the Advanced Photon Source. And they have several things. They look at material science, physics, chemistry, a lot of stuff. Of course, biological sciences is one of the main uses as well, looking at elucidating structures. And so you can see actually on the right, the Advanced Photon Source, the large building on the bottom of that is actually the Advanced Protein Characterization Facility. So they have a direct access to the beam line. So the high throughput structural biology pipeline. So this is a pipeline it's a service, and it's a pipeline that's been refined over several years, supporting several core initiatives. And this is what they wanted to get me, what the head of the structural biology core wanted to get across was our when we talk about high throughput, we talk about high throughput. We wanted to get provide a scale of our throughput. So many of the processes are automated. It's not fully automated, but we have a lot of equipment working in parallel, semi-automated. You can see that like for cloning, they have a capacity of 20,000 expression constructs per year, using several different cloning vectors. Protein expression, a capacity of roughly 10,000 proteins per year, using, you can see the expression system from eukaryotes to prokaryotes. The protein purification, they have several parallel workstations set up in the facility so that they can they have a capacity of roughly 7,000 proteins per year. You can see characterization, and the characterization is 8,000 proteins per year, but it depends on, of course, what the characterization that you're employing. And then with the crystallization, this was kind of cool. They have semi-automated crystallization using several different, again, like several hundred different, I'll call them recipes, but to get some of these more complicated proteins or difficult proteins to crystallize. And they image it during the crystallization process, so you can actually see the crystal form. And that has roughly 7,000. So you can see there's a high capacity for taking proteins, taking a target and going all the way to X-ray crystallography to identify the structure. And so this, of course, is done in the core facility, and then it goes to the APS for structure determination. These are just a couple examples of some viral-related research that was done at the Advanced Photon Source during the COVID pandemic. That was obviously a big issue. So they were with several private sector companies and academia, identifying 65 structures, but not only the structures, but their complexes with their with ligands. That's just an example of 4 of these structures. They also work with Pfizer identifying for Paxlovid that was actually worked on at Argonne, identifying the nirmatrelvir, that's the active ingredient, how that binds to its protease inhibitor, and that was determined at the Advanced Photon Source as well. So they do have expertise, not only working with viral proteins, which can be difficult, but also working closely with the private sector as well as academia. Data management. So this is the next thing that I wanted to talk about is we have a system called Globus. It's out of the University of Chicago, actually runs it. And so University of Chicago actually runs Argonne, And so Argonne and the University of Chicago are very close, and many of their staff at Argonne are also professors at University of Chicago, and that's the case with this. Ian Foster is the lead for Globus, and he is an Argonne employee, but also University of Chicago. So what this Globus system does is it's a scalable data ecosystem that integrates information and functionality across a program, in this case, across APECx. So what's complicated with, everybody wants to use AI and machine learning, but to do that, of course, you need the data. And I think many of the people I talk to spend more time curating data, identifying data sets, curating data, getting the appropriate metadata so that they can actually use that data within a computational environment. So what this does, it allows for all the data from a program to go into a cloud kind of repository and allow metadata and all the other associated data to be structured in a way that it can be used outside of just APECx. So but what it does is it also has several checks and balances for security, who can get access to it, who can pull the data down, how to transfer data. So it's really, it is an ecosystem, but it allows, it ties everything together and allows securely move, share, and discover data. And it sounds simpler than it is, but a lot of people go right to the AI and right to the fun stuff. But really, the front end data has to be, and it should be moving forward. That's how the government should. Everything should be interchangeable, and the data should be able to use outside of a program. So Globus technology, it's been around actually, I think, for close to 15, 20 years. And they actually deploy it at the advanced photon source. Like I said, we have the 60 beam lines around there. I think every one of them has a Globus Connect. And it allows, not only is that a data management, but allows you to execute functions too. So you can actually execute, like at Argonne, if you wanted to do X-ray crystallography, you can actually run it from home. You don't actually have to come there anymore. So this environment just ties everything together and allows the data to be shared within and between programs. High performance computing. So Argonne is one of the, so Oak Ridge, you just heard from Oak Ridge, they are the other leadership computing facility. DOE has 2 leadership computing facilities, Argonne and Oak Ridge. Oak Ridge does have the fastest today. But Aurora on the right there, you see Aurora exascale. So we're number 2, and that's, we only have half of it turned on. So next year Argonne will have, I think it's supposed to be in excess of 2 exaflops. So right now they're stabilizing Aurora and fine tuning it. But for they've been working on it for several years. So for the last several years, they've been working with users to properly change the code, make it so that everything all the codes you can pull down from GitHub and all these other places can work on GPUs in an exascale environment. So they've been working on that for several years. So again, it's turned on halfway, I guess you would say, because of the fine tuning. But so that's the Aurora exascale computer. Overall, the ALCF, it's as a user facility, users can come to Argonne and request node time or compute time, and then you get so much time allocated to you. And there's several different mechanisms for that. And Argonne staff will figure out, work with you to figure out which, I think we have 3 mechanisms, which mechanism is the best. And what's great about it is they also work with each user to make sure that your code can come in, your code can run. How you can execute your code on the exascale environment. And one interesting thing, what I think differentiates Argonne is on the bottom, you see where it says computational biology underneath ALCF. Our biology division, actually all of biology is within our computational directorate. And that was done 25 years ago by our director. And because he could see that biology is probably one of the hardest things to model, and because it's very squishy when you talk to like physics, hard sciences. So anyways, the biology departments are within the computational directorate, and our director and senior management are very bio-focused, I'll put it that way. The vast majority of the compute that's done is related to biology. It's not like we allocate 10% of compute time to biology. Biology is a huge portion. So there's a lot of emphasis and focus on biology. And that shows with a lot of the other programs that Argonne's involved with NIH and other institutes where the Argonne leads in computational biology. And so I guess I'll wrap up, but real quick, I mean, it's very good timing. I was talking to Andy. I forgot to mention our APU's upgrade. So I skipped our advanced photon source is currently down. It's getting an upgrade, and it'll come back online in March. It'll have 500 times the beam power it has now, and so it's going to be it greatly accelerate not only the resolution that you can get, but also the time of acquisition, decrease the time of acquisition. So combining the APSU, the advanced photon source upgrade with Aurora, when that comes online fully next year, it'll be a fantastic resource for everybody, but certainly for this community. Thank you.
Okay, thank you, Dan. And so our last enabler, Danny Dewick from the Vaccine Research Center is going to come up and talk about the work that they're doing in support of the APECx program. Thank you.
Hi, everyone. I am going to talk about a few things that we do at the Vaccine Research Center, and then I'm going to explain at the end how what we do can integrate with what people want to do within APECx. So the Vaccine Research Center is an intramural division of the NIAID. And we perform basic science research to make discoveries supporting the development of novel vaccines and biologics targeting infectious diseases of global public health importance. Here are some of the viruses that we work on. And we've had some recent success, for example, in the field of COVID vaccines and monoclonal antibodies that some of you may have heard about. We don't just work on viruses, we also work on bacteria such as TB, parasites like malaria. We have cancer projects. We do we also work in basic immunological concepts at one end of the spectrum and platform technology development at the other end of the spectrum. What's unique about the Vaccine Research Center as a government institution is that we do everything from discovery to product development and clinical trials. This is the number of people in each building. So we begin in this building here with a basic, and that's where I work, with a basic and translational scientific discovery. We then do process development at our vaccine production program laboratory in Gaithersburg. At our clinical materials pilot plant in Frederick, we actually make the clinical product. Let me just do a laser pointer. All right. We make our clinical product and we use that product in our first in human clinical trials. So we make enough clinical product every year for about 5 or 6 phase 1, phase 2 clinical trials. And then the samples from those clinical trials go back to our vaccine immunology program where we do high throughput immune analysis. So it's this multidisciplinary structure with specialized staff and facilities that accelerates the path from discovery to product development to meet our unique programmatic needs. So in a sense, we're a bit like a medium-sized pharmaceutical company. So that's the VRC. I'm now going to talk about Premise. Premise is why I'm here and interacting with APECx. Premise is a pandemic preparedness program that we started just over 2 years ago. So it's really quite new. It is a pathogen agnostic, that's important, anticipatory reagent and countermeasure discovery program, and it's based at the VRC, right? It's an intramural program. The goal is to establish a preemptive repository of immunobiological countermeasures, immune assays, and pathogen-specific knowledge to accelerate the response to potential pandemic threats. Now, you've heard of the 100 Days mission, from release of the sequence of the outbreak pathogen to having a candidate vaccine or candidate monoclonal antibody, not lots of it that you can give to people, just the candidate. 100 days worries me, to be honest, because Omicron can go around the globe 3 times in 100 days. So our aim is to have stuff on the shelf ready to go as candidates, not a stockpile, as candidates 100 days, 100 weeks, 100 months before the outbreak occurs. So how do we do this? The process is slightly different from pandemic preparedness efforts in the past. I have to disclose I'm not a virologist. I really don't know much about viruses. I can barely remember the names of them. I'm an immunologist, and so we approach this through immunology. So what we do is immune analysis of human cohorts from around the world to detect reactivity, immune reactivity against potential pandemic viruses. And I'll show you how we do that in a second. From that, we deliver as quickly as possible these publicly accessible deliverables. We want to give them away. The mainstay is to make monoclonal antibodies with potential for prevention and therapy. You can also use those as reagents and data resources for early detection and diagnostic assays, and we use them for structure-based immunogen designs suitable for further vaccine development. So this is how it works in practice. This is what we've been doing for the last year or so. That's the population of the globe. And we get samples from focused cohorts and broad-based cohorts. We collect serum, we collect immune cells, that's critical, and we put them in the freezer. At the same time, we do virus discovery. We work with a lot of the virus hunters who are out there. We do a lot of the sequencing for them, and we also mine the databases. And we do that following, we do some down selection following that to design our antigens for our assays. The antigen design nowadays usually starts computationally. Okay, so I'm preaching to the choir here. So we start computationally, and then we make our serological assays, which can be based on peptides, phage display, monomers, multimers, whole inactivated virus particles. It doesn't really matter. We then screen the serum from our cohorts. We're looking for binding. We don't care if it is antigen specific or cross-reactive. It doesn't really matter. We want to identify people who have an immune response that recognizes the pathogen of interest. If that happens, then that triggers our analysis of the immune cells. We identify antigen-specific B cells. That gives us our monoclonals. We can then do functional analysis, and that gives us our reagents and data for early detection and diagnostic assays, our vaccine immunogens available for further development, and our monoclonal antibodies available for further development. And that's where we stop. That's where the repository stops. And then we can give all that stuff away. We intend to everyone else to work on it. Now, the industry standard from a sample of cells to functional analysis of a monoclonal antibody candidate is about 3 months, if not more, and it costs a lot of money. We have a new pipeline at the Vaccine Research Center where we can generate about 800 monoclonals in 7 days. In the last 6 or so months, we've discovered about 25,000 monoclonal antibodies at the Vaccine Research Center, done the functional analysis to all those viruses, some of those viruses, most of those viruses that you saw on my second slide. We take those antibodies and we're embarking on a project in collaboration with another national laboratory that is not represented here today, the Lawrence Livermore National Laboratory. So that's going to be great fun in the future. It's computational optimization of the monoclonals that we have discovered. Okay, I'm going to go through this briefly. This is for the molecular biologists out there. You'll enjoy this. This is our rapid assembly, transfection, and production of immunoglobulins pipeline. So you start with a sample of blood with B cells. You have a probe. You've got to start with a probe because what we're trying to do here is to down select as quickly as possible. So you have an antigen probe, you sort your B cells into 96 or 384 well plates, extract the RNA, you've got the total mRNA from each cell in each one. And from here on out, all of the steps occur in the plates, automated, no bacterial transformations, no plasmid preps. That speaks to the laziness inside me. it spells rat pig, okay? Rapid assembly transfection, it spells rat pig. Rat pig is now a verb. People say, are we going to rat pig that sample? Oh, yes, we are. So this is how it works. We've got the mRNA from the single cell, from a single B cell in each well, antigen-specific B cell in each well. We do some amplifications, we split into two reactions for Ig, PCR enrichment, heavy chain and light chain. We get that product. We then stick bits and pieces on it so that it expresses nicely in a mammalian cell. We assemble all that into pieces. We amplify that, assemble that into pieces, and we end up with an expression cassette, linear expression cassette, that we just do a 96-well transfection with Ig expression and a functional assay. And at the same time, we sequence every single one of those immunoglobulins. So what's important is the timeline. The cell sorting, cDNA and Ig enrichment takes a day, cassette assembly and amplifications a day, transfection and MAB production takes 3 to 7 days. So that gives you a week. One person with one robot, that's Amy, and that's the robot, can make about 400 monoclonals in 7 days with 90% efficiency. If there's a cell in the well, you will eventually get a mill of about 50 micrograms per mil of monoclonal, which is plenty to test. If you want to increase your capacity, you get more robots and more people. It's not that Tim is very, very tall. He's kind of average height. It's just that Amy is very, very small, but very powerful. So premise, we are not a priority pathogen program. We're not a prototype pathogen program. We just have a list of viruses that we're worried about, and it's intelligently called the premise list of bad viruses. We do have priorities, though. The ones in red are the viruses that spread from human to human. The ones in black are the ones that only do zoonotic spread. They're all over the world. We've established this list by going and talking to scientists all over the world, governments all over the world, militaries all over the world, and have asked them, what are you frightened of? And we just add it to the list. And then we make the immune assays. The ones in green are ready. You can see most of them are now, they pertain to the viruses that spread from human to human. These are some of them. We call this the virus world domination plan. As you can see, they're all over the world. So we've established, been busy establishing over the last year, what we call the premise world domination plan or partnership plan. So we have partners all over the world. In green are the partnerships that are established. Pink ones are underway. The orange ones are proposed. And we work really on a kind of lab to lab basis. We don't work with the World Health Organization and big things like that, because there's 5 people in premise. We're really small, we're really nimble, so we can change our list of priorities very quickly. So it's a lab to lab basis, and it works very well. And we discovered when we found out that the government was not going to give us all the money that they had promised us during the pandemic, that we had neither the time nor the people nor the infrastructure nor, quite frankly, the money to do everything for everyone all over the world. So we realized we really had to establish local analytical capacity. So what we do is we go to these labs and we ask them, what viruses are you worried about? What are you interested in? We train them how to do things, and we give them the premise vision of how to approach pandemic preparedness, and they go on and make the discoveries, and they can then make them and give them to their people. Basically, it's a cult. Our mainstay immunological platform is the mesoscale discovery plate assay. This is a fancy ELISA. It's very sensitive. It's very good. It's 96 well plates with 10 spots in each well. Each one can be a virus or even a mixture of viruses. So the childhood respiratory panel has been up and running for a year, and we have some nice projects going on with that. Those are the viruses there. You can get this from MSD now. The regional panel 1, we're doing panels regionally, not by virus families or anything like this, because it makes more sense because we're really a regional network. This is the Africa and the Middle East plate. It's got these viruses here. It'll be ready in a few weeks' time. And then 2024, we aim to have these 2 plates, the Asia and Australia and the Latin America plates, finished. The nice thing about this is that you can easily swap proteins or viruses in and out according to what your needs are. So these are the plates that are going to go around the world for our various projects. how we can collaborate, how Premise can collaborate from within the VRC with the APECx teams. I think the most significant impact we can have is in technical area 3, translational candidate development and clinical evaluation. What we can do can include discovery of monoclonal antibodies by rat pig and library production. So libraries of these monoclonal antibodies for epitope level analysis for the design of the immunogens, structure-assisted design of immunogens using monoclonal antibody libraries. From there, we can help with immunogen selection, which feeds back to tech area 1. That leads on to vaccine platform refinement and testing. And of course, we can do animal model immune profiling. That can include many of the things that we do at the VRC, which includes humoral immune profile, such as binding, FitSeq, which is phase display based assays, neutralization and other functional assays, T-cell response immune profiling by various assays, independent candidate evaluation, that's one of the program challenges, and of course, GCLP accredited bioanalytical testing using our qualified assays, because on the third slide that I showed you, the vaccine immunology program is a GCLP accredited facility. Just to show you in pictures, to go over this again, there's TA3, and the discovery involves monoclonal antibody library production, massive amounts of antibodies that you can use for epitope level structure assisted immunogen design. The optimization includes platform refinement, animal model immune profiling, which feeds with refinement into the TA1 and TA2 model validation and retraining. And of course, the demonstration of this with independent challenge evaluation and qualified assay development. This is the group and a few hangers on. If you have, that's me, obviously, and that is Alison. Alison is over there. So if you have any questions, you can ask me, but really ask Alison. Alison is the one who runs the program. I just show the slides. That's what I got. Thank you.
Okay, so that concludes our, rapid fire discussion about some of our U.S. government partners and the capabilities we are bringing into APECx and for you all as potential performers. I would like to invite Amanda from RTI to come up here. We're going to switch gears a little bit. We're going to talk about vaccine equity and acceptability and accessibility. And then my scientific and technical team is going to come up and we're going to do some FAQs based on what we've heard from you all and some of the more traditional FAQs that come with an ARPA program. And then we'll be ready to break for lunch and we can talk more about that a little bit.
Hey folks, how are you? We're getting closer to lunch, so that's great news. All right, it's a pleasure to be here with you today. My name is Amanda Onwuka. I'm here to talk about vaccine access. A goal without a plan is just a wish. We need to be proactive in designing our strategic plans with vaccine access in mind. And so that's going to be the subject of this conversation today. Again, as I mentioned, I'm Amanda. I'm an epidemiologist by training, and I'm here on behalf of RTI International. RTI has a partnership intermediary agreement with ARPA-H to advise on a variety of different topics, one of which is health equity. And so that's why I'm here with you today. We begin with vaccine confidence. It's sort of the elephant in the room, if you will. As these surveys have been conducted over time, we see that the confidence that folks have in vaccines, trust in scientists, opinions about vaccine safety and effectiveness are diminishing both internationally and then also in a US context. This is important to be mindful of for a few reasons. One, the idea that if we build it, they will come is a fallacy. And so we need to be thinking about vaccine access from the inception or from at the beginning of the process. Second, I just want to posit to you that if I were to ask you all whether you think vaccines are safe, whether you think vaccines are effective, your trust in scientists, your results in this room would differ very much from the national public. And this is important to illustrate because we all bring biases to our work. And it's important to be aware of our biases and then also to integrate various different checkpoints so that we're not just preaching to ourselves, but that the message that we're getting really can reach the broader American public. These opinions about vaccine confidence and distrust in science extend to opinions about artificial intelligence as well. This is a survey by Pew Research Center that was asking folks, what are your thoughts about AI? There's some folks that are more excited than concerns. My guess is most of you are in that group. I'm probably in the middle here, equally concerned and excited. But if we look at the survey results, there are a lot of folks who are more concerned than excited. And some of the issues that they bring to the forefront are, one, issues around data privacy, two, concerns about ethics in government and also ethics in technology, and three, concerns about the lack of checks and balances, and the lack of oversight and regulation. And so this is something that we can be conscious of as we're designing these vaccines that may or may not integrate novel methods. Opinions about vaccine confidence are going to have an influence on vaccine uptake. And we know that there are disparities in vaccine uptake by rurality, by socioeconomic position, healthcare access, race, ethnicity, ability, sexual and gender identity. Literally every subgroup, there are going to be differences in terms of whether or not vaccines are viewed as acceptable and whether or not folks receive them. And this, of course, is going to have implications on incidence in those populations. This is literally what we want to avoid, is that the marginalized get sicker as a result of their disengagement in vaccines. So it can be overwhelming thinking about vaccine confidence, but there's some things within your control that I want to illustrate here. There are opportunities that are within your sphere of influence that can influence vaccine uptake. And so those are some of the things we'll be discussing today. First, are the most important problems in the field, and what are some of the solutions that you're hoping to be a part of as a part of this program? And if we were to ask the general public that, there would likely be disalignment in the values there. This is going to have severe implications, again, as it relates to vaccine access and uptake. And so as we think about the theory of diffusion, there should be alignment with values. And so that's what we're talking about here. The topics that you intend to address should be problems that the public also perceives to be a problem. And the innovations that you're proposing should be attractive and desirable to those populations. There may be some primary or second data collection that you engage in, potentially through patios, customer experience hub, et cetera, to try to get at, does the public perceive this to be a problem, and do we have the potential to really make an impact here? Also, constructing multidisciplinary teams is going to be a huge asset here. We've talked a lot about how to integrate folks with the appropriate technical expertise. I'd like to posit to you that considering engaging an investigator that has access at the forefront of their mind because of the discipline that they come from is going to be a huge asset to your team as they are bringing up these issues as a part of methodological conversations and dissemination and at every stage of the process. You may also be interested in evaluating the potential impact of your evaluation of your innovation on populations. And so this is just one paper. They looked at the impact of appendicitis, lung cancer diagnosis, and herniated discs on a few different economic and health outcomes, including mortality, labor earnings, labor market participation, et cetera, et cetera. And so they were able to identify that these diseases are associated with these outcomes on a global scale, on a macro scale in the US. A similar opportunity could present itself related to vaccine development and disease eradication. So these you may be able to pursue some analysis like this that posits that disease elimination is going to have these effects on the labor market, on labor earnings, on disability, et cetera, mortality. Because of the biases that we all bring to this work, another opportunity that you all have is to create an external steering committee. And this committee could be advising on the desirability of the innovation, the methodologies that you employ, as well as the interpretation of the findings and potential deployment considerations. Disease-specific advocacy organizations are your friend. These folks are going to have more expertise than the lay public, but obviously less technical expertise. And so these folks are great to advise on the desirability of the innovation, on features of the vaccine that are going to be relevant to your development process. Health providers repeatedly in every study are the most trusted source of vaccine information. Integrating those perspectives, particularly health providers that are serving in under-resourced communities, is going to keep both their concerns and the concerns of their patients at the forefront of your minds as investigators. And so we encourage you to be talking to those folks as well. Bioethicists would be a great component to add to your team, specifically as we are discussing the employment of methodologies that are new in the public sphere, and that the public has demonstrated concern about. Integrating a bioethicist into your either research team or external steering committee could have great advantages in terms of how to translate your findings to the public. And then finally, just other multidisciplinary folks that have a different eye towards what is important and different considerations that you can be making. The goal here is not for people to merely advise, but for there to be a feedback loop whereby their advising then informs you all as investigators. Maybe there are some modifications in terms of your design, your approach, and then you take that back. Maybe you don't want to integrate.
Okay, apologies everyone, we had some network issues. We stopped our presentations as soon as we saw that we were disconnected, and so Amanda's going to pick us right back up.
All right, folks. So we're talking about the TPP, its ultimate importance. I just wanted to underscore a couple things. When we think about cold chain, especially, cold chain is not merely an issue that's of international importance. Even within the US, we observed that rural communities have less access to the refrigeration units, et cetera, that are required for cold chain. Also related to resealable valves, this is another opportunity that to reduce waste and increase access. And so I encourage you all to be creative and to sort of decrease the thresholds for engagement. Another opportunity I wanted to talk a bit about was the effectiveness against severe disease versus moderate disease. A lot of what's driving vaccine uptake are perceptions, right? It's not merely objective data, it's perceptions of effectiveness, not actual effectiveness. And so if, for example, we have a vaccine that's protective against severe disease, if I get the disease, I get a moderate case of disease that requires multiple healthcare encounters before being resolved, I may perceive that the vaccine did not work when in fact it was just it never said it would be effective against moderate disease. And so this is something to keep in mind, that if those sorts of narratives are perpetuated, that could undermine what we're trying to do here. So please keep that in mind as you're developing your TPPs. As we think about health equity, that's like the key driver behind this conversation today. One of the realizations that we have is that to be a leader in science and technology requires a vast amount of resources and opportunities that were not always within our control. And so building capacity within communities or environments that may not have had the same access to opportunity is a critical component here. And so the data sharing requirements that are in the BAA, we ask that you think about how to actualize the minimum data sharing requirements, and then any additional metrics that you might be able to integrate as well, given the nature of your work. Also subcontracts, we ask that you consider how might you build capacity using subcontracts. These are all opportunities to promote health equity. And so as I think you're hearing a lot of the same themes today, which is one, talk to people outside of your area of expertise, and to think about the end at the beginning. And so as we think about deployment strategies, we ask that you, you can see this. We ask that you integrate in your proposals a demonstration of barriers to vaccine uptake and disparities in vaccine uptake with a language and tone that respects individual autonomy. AI is new. Individuals may or may not have a good understanding of the research methods that are involved in your work. And so that is legitimate. It's a legitimate concern for someone to be concerned about some of the methodologies that are used. And so describing them with language that respects individual autonomy is going to help us move the needle forward. Also, you may consider scientific inquiry that will support deployment eventually. And so whether that be surveys or focus groups where you can really get an understanding of the desirability of the innovation, features of the innovation, features of the vaccine that are going to promote uptake. This is some of the work that you can be doing concurrently with development that will both inform your research process as well as inform dissemination and communication strategies. There are now many known best practices for vaccine communication. And there are a couple that I just want to mention briefly. One, everyone is not vaccine hesitant. Vaccine hesitancy describes one segment of the population who wants to know more. They're skeptical about some of the methodologies and maybe a bit resistant, but they're not the same as vaccine apathetic individuals who are disinterested and haven't given a lot of thought to it. And they are not the same as folks that are poorly reached, and they are not the same as active resistors. And so as you think about how to communicate to these different audiences, we encourage you to think about them as different audiences, and different communication strategies will be appropriate for those different folks. Another thing I just wanted to bring to your attention is that we know that misinformation is propagated maybe more faster than the truth. And so you can anticipate some of that misinformation and begin to craft strategies to mitigate that in the development phases. And I think some of the community-based research is going to support that effort. I think this is almost, we're getting to the end here. The FDA has published a lot of great guidance on clinical trials and how to make sure that they're representative of the general public. We encourage you to reference that, integrate their suggestions and adhere to those, and to give some thought to both how to plan for recruitment, both in your clinical and preclinical stages. And so in addition to the technical characteristics of your proposal that you'll be submitting, there are other metrics by which your proposals will be evaluated. And these are some of those metrics. One, does the proposal demonstrate an understanding of the burden of disease in vulnerable populations? Does the proposal identify a multidisciplinary team? Does the proposal describe an intention to engage with external and multidisciplinary experts? Do they identify a target product profile that prioritizes accessibility? Do they provide a plan for data sharing? Do they identify minority-owned or minority-serving institutions to partner with? Acknowledge barriers to vaccine uptake with a respectful tone? Describe any intended scientific inquiry to support deployment, including a potential communication strategy? And finally, describe steps to gather and recruit a representative population. It is our hope that these efforts, the efforts of APECx, the ARPA-H team, and also your efforts yield amazing, transformative, innovative products that change the world. But they will do so only if people use them. And so considering vaccine access from inception, from design, is going to increase the opportunity for you to really survive in the wild. And so that's the message that we're hoping to communicate today. Again, my name is Amanda Onwuka, and I'm happy to take any questions. Thanks.
Great. Thank you so much, Amanda. And so, she really highlighted really the ethos that we have at ARPA-H, which is part of our mission statement, increase better health outcomes for all. And so I think these are not suggestions, really. I mean, when you look at the BAA and the solicitation, there are metrics and requirements built in there, right, for programs. And so this is incredibly important stuff. And like Amanda said, if we're just building really cool tech stuff and no one uses it, we're not going to meet the goals that we want out of the APECx program. Okay, so I know we're a little bit behind with the TechLit, but I think the FAQ portion is incredibly important. And so we're going to go a little bit into our kind of lunch time with that. But I'd like to invite up my kind of technical team to walk through some of the most frequently asked questions that we've heard throughout this process of talking to you all and your potential partners, and then kind of set the stage for the rest of the day.
Hi, everybody. My name is Paishan. I am a contract technical support for Andy. So I'm just gonna go quickly over some of these questions. So budget question, how big can our program be and what is the overall budget for APECx? So we will not provide any budgetary details for APECx or a target number for your budget. No budget ceiling has been established. Offers are encouraged to propose realistic, justifiable, and reasonable budget that is aligned with their proposed solution. ARPA-H discourages strategies such as using only junior staff or proposing only low-risk ideas to cut cost. Can you explain the rules around salary cap in our cost proposal? Since the 1990, Congress had legislatively mandated a limitation on direct salary for individuals under the NIH grant and cooperative agreement awards. The Consolidated Appropriation Acts of 2023 public law restricts the amount of direct salary executed executive level of the federal executive pay scale. Effective January 1st, 2023, the salary limitation is roughly around 200,000. So this means that the maximum of any one person in 1 year for any labor category of any level effort should be roughly around that number. So teaming specifics, do we have to have partner from industry, academia, or something else? No, teams can be any participant from any mixture of institution. Can one group be part of multiple proposals? Can one person be part of multiple proposals? Yes, a group or person may be part of 2 proposals, but cannot be on the same the prime proposal on both. Is there a limit to how many institutions can team? No. But be sure to include detailed budget information for each institution as per the R&D solicitation or the BAA, and include in your proposal and program management structure that explains how the teams will be coordinated. So more teaming questions. Can we have multiple PIs? No, there can only be one PI and the rest should be co-investigators. Can we change the teams partway through the program? Teaming changes may be allowed with ARPA-H's approval in response to specific needs arising from program's progress. Requests will be dealt with on a case-by-case basis. Will you establish teams for us? I'm looking for a partner to complement our capabilities. We're happy that you are looking to assemble a diverse, capable team, but we will not be making any introductions for you. But we encourage you to talk to other people at Proposers Day today, especially during a poster session, and/or use our teaming website where you can submit your profile and find a contact information for potential teammates. Yeah, that is listed on the APECx teaming website. So here are some guidelines for some international proposers. Are foreign entities eligible for an award? ARPA-H will prioritize awards in accordance with the public law number 117-328 under the section 499A of the PHSA. Without limiting foregoing, ARPA-H will prioritize awards to domestic entities such as organizations or individuals that will conduct funded work in the US. However, non-US entities may participate to the extent such participants comply with non-disclosure agreements, security regulation, export control laws, and other government statutes and regulation applicable under these circumstances. Non-US entities are encouraged to collaborate with domestic US entities in number of cases no cases will award be made to entities organized under the law of covered foreign country as defined in section 119C of the National Security Act of 1947, or entities suspended or debarred from business with the government. Foreign entities may be subject to clearance review prior to beginning work on an award, whether as a prime or sub. Where foreign entities as sub, awards may be made with a foreign support restricted under the clearance process is completed. Okay, one last thing for me is guidelines for government proposals are federal entities or federally sponsored entity eligible for award. Federal entities and federally sponsored entities such as government national laboratories and federally funded research and development center, university affiliated research center, military educational institution, etc., are not eligible for award under the announcement. However, ARPA-H is committed to working with its federal partners. Federal partners interested in working with ARPA-H on this program should contact us at ARPA-H, APECx at ARPA-H.gov to discuss supporting effort. Okay, I'm going to get Hajan to present next.
Okay, nice to meet you, everybody. I'm another tech leader. My name is Hajan Park. So I'll go over, is that, yes. So these are basically rules for the, play. So just skip a couple of these. So those are like submission timeline and feedback, most important part. So here, about the feedbacks and the technical concepts you're trying to communicate, I think after today or beyond the sidebar, we won't be able to do that. So so just keep in mind. And then the abstract dues December 1st. That's a 4-page abstract. So you'll submit. So that's the only way to communicate with us, ARPA-H. And then December 8th. So that's the so between the December 1st and December 8th, we'll go through the review process of your abstracts, and then we'll give you a feedback letter and invitation for full proposal if you're accepted. And in the abstract, there are 3 outcomes. So one is you're not selected, and second, you'll be selected and go for a full proposal. And the third, we may contact you if we have some questions about your abstract. So roughly after 6 weeks, so you'll finish your proposal and submit. So full proposal due is January 19th, and then between January 19th and March sometime, we'll go over the, we'll do the review process, and then the notice of award will be made on March 24th around the time. And then between March and April, we'll go through, if you are selected, we'll go through contract negotiation process. And then April will hopefully kick off the program. All right, so, and we'll do that.
And so, hello, everyone. My name is John Shannon, and I'm also a technical CETA for APECx. And so a few other questions we have. One has been, who will actually own the intellectual property? And generally speaking, the government doesn't take ownership of any IP. And for those cooperative agreements, the performers will retain their IP. And then for the OTAs, they're a bit more flexible, and the IP will be subject to negotiation between the proposers of ARPA-H. One thing to note is the Bayh-Dole Act does allow the government to retain or take ownership in certain situations, but those are extremely rare, and it's generally if there's some compelling national interest. And so at minimum, the organization does want to ensure that IP restrictions are not keeping any breakthrough technologies from the people who can benefit from them. And so for a few specific questions that we've gotten from our R&D solicitation, and so we've been referring to that as BAA through most of the day, and they're the same thing. But one is the R&D solicitation refers to 3 technical areas. Do I need to address each of them? And so Andy's covered this, but we just really want to make sure everyone's aware that submissions can either address all 3 technical areas or TA2 alone. So if we have TA2 alone, they'll have a unique set of milestones and deliverables. But if we get any TA1 or TA3 alone submissions, those will be deemed as non-conforming and they will not be reviewed. So just keep that aware. And another thing is we are expecting that all proposal teams will actually have the expertise to collectively achieve the goals that they are applying towards. Another common question we've had is, do we have a target pathogen? And so does APECx have a preference for a specific viral target? And we do not. So however, we do not want to receive proposals that focus on families that are already well funded or extensively studied. So that includes influenza, SARS-CoV-2, and HIV. And then another thing to keep in mind, and Amanda highlighted this, is proposers are really encouraged to consider the equity aspect of viral disease targets in their selection. And so now just a few quick metric questions. So the requirement timeline seems aggressive. Do you really intend us to hit these metrics? And so broadly speaking, we hope you do. We really want you to work hard to meet the goals of this program. However, we realize that in specific cases that may not occur. And so as long as there's metrics that show some quantitative improvement compared to the current best practice, we will consider that as success. And then what if I do not meet the metrics at one point in my project? Will you stop my project? And as Amy mentioned in her talk, like some performers will not make it through the entire thing. And our program is separated into those two phases. So there's phase 1, that's 3 years, phase 2, that is 2 years. And so all performers will not continue to that phase 2 stage. And, you know, there's not rigid go, no go criteria. Instead, it's looking at whether or not sufficient progress has been made and which performers will proceed through that down selection phase. And so one thing to note again is all of this material will be available on the APECx website, and including the slides and recordings. And the final thing we really want to make sure is read the R&D solicitation, because many of the questions that we've heard here today and probably you guys still want addressed are going to be in that R&D solicitation. And as Jennifer Mack mentioned, like follow that more than anything that's been said here. And so there will also be an FAQ session or section on the APECx website that should hopefully be put up in the next day or two, and that will list any questions that we've had come in and answers for those. And if for some reason your questions are not answered in the R&D solicitation or that FAQ session, please email the apex@arpa-h.gov email, and we'll get back to you with that. And with that, I will ask Andy to come up for some final remarks.
Yeah, great. Thank you, John, and thank you, team, for going through those FAQs with everybody. For all of those who are virtual, we really appreciate your attendance and your participation. Thank you so much. We're going to work to share the rosters of the people who have attended Proposers Day. We have a teaming profile site that was mentioned earlier, but it is a benefit for those of you virtual to know who all has been participating in these discussions. And so we're going to work to facilitate that. And that kind of concludes our morning session, which is the hybrid portion. For the rest of the day, we're going to be just in person here, and that includes in-person sidebars and the poster sessions. And I mentioned earlier that for those of you who aren't here in person to have a sidebar discussion or who didn't have the opportunity to sign up, we'll be sending an email after the meeting wraps today with sign-up sheets for time slots next week to have virtual sidebar discussions. So opportunities to ask questions and get those answered if we haven't done that already today. Okay, so I kind of ran through the rest of the day already. We're going to break for lunch now. I'd ask that for the folks here to be back at 1:30. There's a bunch of restaurants right around the area, so there's a lot of things that are close. And then please set up your posters. And for those of you who have a in-person sidebar, we'll be doing that right down the hall, and we have a schedule out there in the hallway as well. So thank you everyone so much. Really appreciate it. We are incredibly excited about everyone who's participated in the meeting today, and we are anxiously awaiting submissions in a few weeks. Thank you very much.